is important in that process. I mean, there's been more papers out recently that describes the mechanism, but I think it's clear that by doing a meta-analysis that some groups, including gastric and some renal, there's something about, and then they seem to have a specific immune evasion mechanism. So we're now delving into why some groups have more hyperprogression than the other. So this becomes an ethical issue because, sometimes we blindly put them on immunotherapy trials and then they just end up having lots of hyperprogression and die sooner. So our group is focused on developing a marker for hyperprogression. What predicts marker? So because first principle is do no harm. If you're a physician, you don't want to give an agent that will make them die sooner. So we are doing proteomics, some cytokine, or just looking at tumor microenvironment and using the H&E based AI to look at the images of the pathology slides and really combine all these factors to predict who are going to be the hyperprogressors. But understanding the interferon gamma pathway seems to be the key, according to what Min-Jung Kim has been reading. So that's why we invited the expert, Dr. Taegong Kim, to pick his brain. So with that, I'll let Min-Jung Kim talk.
Thank you, Dr. Che. I'm sorry for the delay.
Okay.
I had to shift the order of my presentation a little bit. First of all, it's a little bit frightening to present in front of Dr. Taegong Kim, especially after doing a presentation, a journal club on his review. I'll share my slides.
Some of my members sometimes cry during the lab meeting.
Pardon, sorry, I couldn't.
Some of my lab members cry during the lab meeting.
Oh, that's okay. That's okay.
I'm kidding.
I'm welcome to criticism. So let me share my slides. Okay. Let's see. I don't know where the. So just to give a brief background on what hyperprogressive disease is, for those of you who are not very familiar, after the advent of immunotherapy, we've been experiencing novel patterns of response. So before the era of immunotherapy, we're most familiar with progression, which is when the patient develops some sort of resistance to the therapy that they're receiving. But with immunotherapy, we started observing two new patterns of response. One of them being pseudoprogression, where the patient seems to have experienced a growth in the size of their tumor, but the tumor shrinks in size again when the patient is reevaluated at a future time point. In contrast, we've also experienced a more severe and critical form of response called hyperprogression. And this phenomenon is not yet uniformly defined, but basically it's characterized by an accelerated growth of the tumor, defined usually by tumor growth dynamics based on calculations that are taken from the RECIST measurements of the tumor at the first evaluation after starting immunotherapy. So that was the very, very minimum explanation of hyperprogressive disease, given the time limit that we have today. Here I'm presenting the forest plot from the meta-analysis that I've been working on with other people from the lab for the past year. And as you can see, even though the confidence intervals are wide, when we run logistic regression, for the tumor type, we found out that the odds of developing HPD is higher for patients with advanced gastric cancer compared to renal cell carcinoma. This was the biggest difference in the odds ratio. And a significant difference was also seen between renal cell carcinoma and hepatocellular carcinoma, mixed or other types, and non-small cell lung cancer. So given these results, the next step then was to try to brainstorm what might explain these differences in the HPD incidence that we have been observing. And so the next step was looking into what are putative mechanisms of hyperprogressive disease that are known. And so I won't be covering all of the mechanisms here, but here I bring up a figure from a review from 4 years ago. And this is just a summary of some of the mechanisms that are known. Today, I'll be covering mostly the ones that have been demonstrated in patients instead of just in vitro or in vivo models. So broadly speaking, there are 1, 2, 3, 4, 5 mechanisms. Treg cell expansion, T cell exhaustion, modulation of pro-tumorigenic immune subsets, aberrant inflammation, and oncogenic pathway activation. So now I'll be going into a little more depth for each of these. So for Treg subset expansion, a study from 2018 by a group in Japan on advanced gastric cancer patients showed that there was a marked increase in effector regulatory T cells with proliferative capacity in a TIL analysis. And in another most recent study from Korea, when they looked at the peripheral blood of NSCLC patients, they saw a significant increase in this long name here, subset of Treg cells, which are known to suppress effector T cell proliferation. And another study from a group in Spain in also non-small cell lung cancer showed a significant correlation between HPD patients and the expansion of highly differentiated T cells. And in terms of prognosis, it's more preferable to have less differentiated T cells. So this was related to poor prognosis in their paper. Moving on to T cell exhaustion, a study from 2016 in non-small cell lung cancer patients. They analyzed the pleural effusion of patients who basically had rapid progress. This wasn't necessarily hyperprogressive disease, but they progressed rather quickly. And they were able to find out that there was an overexpression of TIM3 in tumor infiltrating cytotoxic CD8 plus T cells at the time of disease progression. And interestingly, the presence of TIM3 was significantly correlated to the duration of PD-1 blockade. And another study in 2021 in a subset of NSCLC non-small cell lung cancer patients, they were able to observe an expansion of T cell immunoreceptor with IG and ITIM domains. It's short for, in short, it's TIGIT, positive subtype among PD-1 plus CD8 plus T cells. And both of these are markers of T cell exhaustion. The third one is the modulation of pro-tumorigenic immune subsets. So a study in 2015 in a group of melanoma patients showed an upregulation of IL-10 receptor expression by PD-1 high tumor antigen specific CD8 plus T cells upon PD-1 inhibition. This study, though, was conducted ex vivo. So they basically took the tumors, they basically took the blood samples from stage 4 melanoma patients who had not been treated with immunotherapy, and they incubated it with PD-L1, and they measured how the expression of IL-10 receptor changed after this incubation. Next, another study in 2017 showed increased levels of angiopoietin-2 in serum upon PD-1 inhibition in patients with, again, advanced stage melanoma. And interestingly, there was an increased infiltration of CD68 positive and CD163 positive macrophages in the post-treatment tumor biopsies of ipilimumab plus bevacizumab patients with increased vascular angiopoietin-2 expression. And what's notable about these two studies together is that both IL-10 and angiopoietin-2 are included in the innate PD-1 inhibitor resistance signature. So this was a signature group or a signature of genes that were previously associated with resistance to another treatment. I can't remember what it was currently right now off my head, but they found out that this was also associated with PD-1 inhibitor resistance. And on a similar note, we're still on the, oops, sorry, please ignore. Oh, wait, please ignore this, but on a similar note, now moving on to interferon gamma, a study in 2013 showed that interferon gamma driven upregulation of inhibitory molecules was involved in Tregs, let me restart. So they observed interferon gamma driven upregulation of inhibitory molecules that were involved in the Treg cell differentiation, such as IDO. And so with that overview of the putative mechanisms, that's when I started looking into basically reviews on the immuno-oncologic environment and came upon Dr. Taegwon's review. And I can't go through all of the slides from last time, but just taking one of the slides, some of the mechanisms that he described can be summarized into the loss of function mutations in Janus kinase 1 and 2. And this is important because the activation of JAK signaling is required for cancer cell death, and it's mediated by interferon gamma. And secondly, another loss of function mutation was a truncation in the beta-2 microglobulin gene, which led to decreased expression of major histocompatibility complex class 1. Another mechanism that he described in the review was the upregulation of other inhibitory molecules like TIM3, which I've described before. Deficiencies in the presentation of tumor neoantigens, such as the downregulation of HLA expression. And lastly, he touched upon how prolonged interferon signaling induced by anti-PD-1 therapy or CTLA-4 blockade could be a mechanism in which there was an induction of STAT1 related epigenetic changes resembling features of resistant tumors. And when they characterized cells, they found out that there was augmented expression of interferon stimulated genes and ligands for multiple T cell inhibitory receptors. The reason why this last mechanism caught my eye especially was because I had previously read another paper on the overall immune landscape of cancer from 2018, in which the goal of this research group was to characterize the tumor immune microenvironment across 33 cancers. And they based their study on data from the Cancer Genome Atlas. Briefly summarizing what they did, they basically took 160 signatures that have been known to be related to immuno-oncology. They then did signature cluster modeling and was able to categorize it into 5 representative signatures. namely interferon gamma, TGF beta, macrophage, lymphocyte, wound healing. They then did cluster analysis of 30 basically solid cancer types based on these 5 signatures and came up with 6 immune subtypes. And these are the 6 subtypes that are presented in their paper. And here is the interferon gamma dominant subtype. Tumors that basically belong to this category include highly mutated BRCA, gastric, ovarian, head and neck squamous cell cancer, and cervical tumors. And it's characterized by the lowest macrophage to lymphocyte ratio, the lowest Th1 to Th2 ratio, a naturally high proliferation rate, a high intratumoral heterogeneity, and highest M1 to M2 polarization, and highest CD8 T cells. So these 6 subtypes, immune subtypes that I've introduced here, are also presented here. And what they basically did was they looked at each of the tumor types from the TCGA data, and they characterized how much each immune subtype basically comprised each of the samples in the TCGA data. And when we look at the HPD incidence from our meta-analysis, as I introduced before, the incidence was highest for gastric cancer, hepatocellular carcinoma, lung cancer, and lowest for renal cell carcinoma. And just by naked eye, what I did was I qualitatively compared each of these groups, which I've indicated here with an arrow. And if you just taking it simply, if you look at, this is stomach adenocarcinoma or advanced gastric carcinoma. If you look at this one and look at this one, you can see that in gastric cancers, the C2 or the interferon gamma dominant subtype takes up the majority. Whereas if you look at renal cell carcinoma here, kidney renal cell, the inflammatory subtype comprises most of the tumor samples. Going back briefly to the immune subtypes, the inflammatory subtype is characterized by a low proliferation rate, and it has the most balanced ratio of macrophage to lymphocytes. And then taking those 2 stark comparisons aside, if you look at, for example, let's see, if you look at lung cancer, which is represented here in the middle, you can also see that the interferon gamma subtype has a pretty substantial portion in terms of their immune subtype characterizations. And so the questions that we had was, how is this interferon gamma dominant immune subtype? How would it be associated with a higher risk of hyperprogressive disease? That's what we've been trying to brainstorm in the past few weeks. And we're trying to look into what properties of interferon gamma signaling may contribute to adaptive resistance. So with that, I'll open it up to discussion. And please feel free to make me cry because yeah, I really didn't have, I feel like I didn't have enough time to prepare for this, and I feel like it was a rather poor presentation. So yeah, thank you for coming, visiting us during lab meeting when you're super busy.
Thank you, Minjung. So Dr. Tae-Gon Kim, any any comments, or what is your thought based on the findings that she just presented?
That is great observation, to be honest with you. And then it is, I'm kind of impressed, Minjung, and then you, given your based on your stage of the training, and that it is very hard to understand the entire biology, and then also the statistical analysis. I'm kind of impressed, and then I'm pretty sure the Dr. Chen, they guide you pretty well, and then even though you're working hard, and then keep it up, and then I think as I told you before, Minjung, by the tags that your future is really bright. I hope to continue to work harder. In terms of the by the way, this is Ashley.
I actually,
I met with her for the first time today. But okay, so in terms of hyperprogression, and then it is we don't have any clear evidence of that. Biologically, I don't know it makes sense. But I see some phenomenon in the animal model. Some tumor, for instance, this is my view, and then don't just take all my opinion is very personal, okay? So let's say I inject, especially the non-immunogenic tumor, okay? So immunogenic tumor, particularly melanoma, and then let's say some of the lung cancer is from the heavy smoker, some of the renal cell carcinoma, but renal cell carcinoma, we don't know why it's very immunogenic, to be honest with you, but it is very immunogenic. Some of the MSI high colon cancer, their immune signature is predominantly T cell based. Okay. No matter what, even though there's something pseudo progression or whatever it is, it's all based on the T cell. Very rarely some myeloid, but most likely T cell. Let's say some melanoma is malignant melanoma patient is resistant to anti-PD-1, but there are a lot of T cell in the microenvironment. But the people believe that T cell is not antigen specific now. That's most likely bystander T cell. So meaning it's all about the T cell, except some tissue-based metastatic cancer. Then it's different story. Okay. But some non-immunogenic tumor, let's say in our case, in our mouse model, there we have a very non-immunogenic melanoma, B16F10. We inject this tumor in the mice and inject the PD-1 or some other checkpoint. Sometimes the size is getting bigger compared to the control group. So we don't know why. So two hypotheses I have is every single checkpoint antibody behave a little bit different fashion in the context of the tumor microenvironment. For instance, even though we believe we call the PD-1 antibody, PD-L1 antibody, is an immune checkpoint blockade, right? But sometimes it's not the blockade. It is sometimes a stimulate, okay? Especially PD-L1. PD-L1 antibody, sometimes the PD-L1, we believe this one is a ligand, but sometimes it behaves as a receptor. And I still believe that if the PD-L1 antibody is engaged with the PD-L1, sometimes receive the signal and then get some activate the signal, and then the tumor can proliferate. So the one question for you, Minjung, you may need to look at the hyper progression is the more often occur in the PD-L1 group or PD-1 group. Let's say Nivo group or atezolizumab group or something like that, and then see what happens. Okay, that is the one thing I want to know. based on your model, I don't believe that your model number 1, 2, 3, maybe especially 1, 2, and then most often we see the consequence, we don't see the cause. For instance, if we see the microenvironment, there are a lot of Treg. We don't know this is a causal cause of the hyperprogression or consequence of the hyperprogression. Well, we don't know what actually caused this immune signature. Okay, and then interferon gamma theory is very interesting, but we should have invited Daniel.
Oh, we did. He said he couldn't.
Yeah, I
recall.
know. This is California guy. And then, so his observation on one of his cancer discovery paper is suggests that continuous interferon is not necessarily interferon gamma. I believe he used a type 1 interferon, based on my understanding. It's a continuous stimulation interferon signaling in the cancer cell actually change the epigenetic signature in the tumor. So there are a lot of change in terms of the interferon gamma signaling, JAK signaling, also the hypermethylation, etc. Actually make a tumor is very resistant. I believe that actually make a tumor is very, very resistant to any immune signature. I'm not sure this is the hyperprogression is another consequence of the immune, adaptive immune resistance, or by itself is getting a stronger the mutation or whatever, and then make a tumor is more aggressive. We don't know. So this is a totally theoretical speculation. So if you're writing up your manuscript, you have to be a little bit careful about your wording, because it is a lot of suggestion or speculation instead of more mechanistic study. So that is the two comments. I don't believe number 1, number 2, the T cell exhaustion, and then the Treg theory is to make the tumor getting the more bigger and the induced hyperprogression. That is not the right. But I like number 3, maybe the macrophage things, M2 macrophage is a more very, very, very reasonable theory. And then I still believe the number 4 is one of the primary mechanisms. But interestingly, the list of the tumor you show, like a stomach cancer, what is the lung cancer, and then what is the another one, like something?
Hepatocellular carcinoma.
Hepatocellular carcinoma. What else?
Uh,
I think you list 4, right?
there,
Renal was the opposite.
Okay.
Yeah, renal was low.
Okay.
And,
Okay.
yeah,
Okay.
gastric.
Anyway.
Yeah,
Yeah.
liver was high. Yeah.
Yeah, lung and stomach and what else? Hepatocellular carcinoma. Interestingly, 3 location is there are so many tissue resident macrophage. So yeah, that's very interesting, because lung has a tons of macrophage. Many of macrophage is tissue resident. Tissue resident macrophage versus conventional macrophage. People call in the like an immunologist. I call these the macrophages bone marrow derived macrophage. Okay, there are 2 different macrophages. One is a tissue resident macrophage, or the other one is a bone marrow derived macrophage. But liver, we have a tissue resident macrophage. Minjung, what can we call tissue resident macrophage in the liver?
oh, Cooper cells.
Cooper cell. Good job, you passed. Cooper cell. Okay, and then we have the long tissue resident macrophage. It's a very important role, the tumor immune evasion, but no one knows what exactly the long tissue resident macrophage is doing in even microenvironment immune evasion. We are studying that right now. And then third one is basically a stomach. If you look at the stomach, there's so many macrophages. And then this is so dynamic. Whenever you see the progression of some tumor, like early stage stomach cancer to later stage stomach cancer, these dynamics, it's just so beautiful. And so all the things macrophages is very important role. But if I were you, in addition to the interference gamma signature, maybe you can see the M2, dominance of M2 is one of the most important observations. I don't know how the PD-1 stimulate the M2 and then in the tissue-dependent manner, I don't know. But that is one of the primary key to explain the hyperprogression. Okay, I will stop the comment. The problem is for me, once I start talking and I can't stop.
Thank you. Minjung, do you have any more questions or comments for Dr. Kim?
There's so much jotting that I've been doing right now. I think I'll have to go back and read it. And if I have any more questions, yeah, I'll contact Dr. Kim and ask him. But thank you so much.
No problem. It is in terms of biomarker it will be very, very tough to find because of many different reasons. Because first of all, we don't have a good control. Meaning, let's say somebody who doesn't respond to the checkpoint, we don't know that is a hyperprogression or just not respond to anti-PD-1, right? So for me, immune signature is the two category is very hard to distinguish. So only way we can distinguish the microenvironment, but still it is scientifically, I don't know, is clinically maybe we can find one good biomarker, but biologically it is very tough because we cannot control every single confounding factor. For instance, immunogenicity, and then some of the dominant gene mutation, for instance, EGFR mutation in the lung cancer is a typically non-immunogenic tumor. And then compared to stomach cancer, I don't know, HER2 positive one is immunogenic, non-immunogenic, people argue still. And then liver recently approved the CTLA-4, the PD-1 combination, but to be honest, we don't have any clear mechanism why this combination working. So this lack of the mechanism and lack of understanding makes us very difficult to find a biomarker at this time. But if Dr. Che is in addition to probably instead of blood, maybe microenvironment study is very important to address this question. I don't go with the genomics. This gonna be. I'm pretty sure the p-value is not that significant, but there is a new technique maybe you can consider called the nanostring. It is a pretty. I use the nanostring in the mouse tumor. The tissue it's very good, but it's not super expensive. So that's why I suggest you to use the nanostring.
Yeah, in the DART trial, we used nanostring. So we got
good.
signature. Well, anything. So going back to the interferon gamma theory, and the continued stimulation can be detrimental or make tumor grow even faster through whatever the internal mechanism that can be. What is, I guess, do you agree with that classification that immunity paper showed? And then there, I guess the interferon gamma pathway dominant group, do you think that has to do with the continued stimulation, or do you think that's different story? That's a
This is
different.
a different story. This is a little uncomfortable because the Daniel's paper
Yeah,
then is Tony's group.
yeah.
They believe that it's clearly type 1 interferon.
Mm,
Type 1 interferon is interferon alpha, beta, those things. Okay. Gamma is interestingly usually stimulate and then induce the expression of the PDL1.
mm,
And then those category of group usually respond to anti-PD1 pretty well.
mm.
So, but who knows, the interferon gamma behave as same as the type 1 interferon. That's why I want to invite Daniel. Because he is basically he completely changed his scope to epigenetics.
Mm,
So I'm not sure he used the interferon gamma,
mm.
but maybe I'm not sure he's coming to CC, but maybe
No,
we
he
can
said
do-
he's not. But
Mm.
what I'm thinking is maybe Tuesday, 10 o'clock, or if you have time, Dr. Kim, I'm thinking of inviting him for like 10 o'clock. Will be like 8 a.m. So he should be okay. Yeah.
Yeah, I think so.
Yeah. So I'll have Minjung coordinate that with that Dr. Daniel Kim to talk a little bit more about the interferon gamma, the alpha, and the paradoxical stimulation, all that story. Because I think we feel like we need to learn more.
And the
Uh,
interferon usually make a macrophage is more M1, like inflammatory macrophage, typically anti-tumor effect.
mm,
So that's why we don't still understand the tumor microenvironment clearly.
mm,
It is very tough, but but that's why that we are doing the research and then to better understand the microenvironment. This study is very important.
mm. Yeah. So I guess in the
Please,
future,
yeah, please check the anti-PDL1 versus the PD1 is any different in terms of hyperprogression. Especially the macrophages, a lot of the PDL1 expression.
mm. Makes sense. Yeah. So yeah, I mean, the one reason we're delving into this is just like you said, utilizing that nanostring or some transcriptomic profiling. Can we identify a group that will have higher chance of developing hyperprogression? So again, do no harm, and we feel like rather than randomly assigning people on combination immunotherapy trials, understanding the risk first would serve better for our patients. And there are, like I said, M1, M2 shift in hyperprogression research. It's been a field that's been emerging. So I do agree with Dr. Kim that might be the key phenomenon happening here. And I've talked to many oncologists, and most of them believe in hyperprogression because they've seen it clinically. But it's the laboratory immunologists still don't believe.
So I'm neutral, but one thing I also want to know is typically the Nivo
Mm,
and then Pembro, they actually modified FC
mm,
to the human IgG4.
mm,
Human IgG4 is among the IgG, it is the least ADCC effect. least ADCC effect. But I'm not sure atezolizumab or just more the PD-L1 antibody. If PD-L1 antibody is IgG1, I still believe, let's say the PD-L1 expression, even the T cell we found the T cell PD-L1 expression as well. So if it is a depleting antibody, let's say the theory is that the PD-L1 antibody deplete all the immune population somehow, and then the tumor is hyperprogressed. That's another least probable, but however possible explanation.
Yeah, I think I've seen PD-1, PD-L1 subgroup analysis. I haven't seen difference, but that might be old study, but we can update that. Yeah, but I can find the paper that talks about the FC portion of the each therapy, and when they modulated the FC portion of the antibody, the hyperprogression animal model showed no hyperprogression. So they believe that the FC portion stimulating M2 was the main key. And there was a paper that I liked the most, and they did experiment with EGFR and all that. So yeah. But you know, the interesting thing is if you add chemo, then you know all of the hyperprogression disappears. So chemo immuno combo is, you know, that you get so, and on all the immunotherapy trials, you have the chemo versus immunotherapy, you have always crossover for the progression-free survival, because initially you have hyperprogressors always with immunotherapy group. But then with the chemo immuno trial, there is no crossover. So that's been a consistently reported phenomenon as well. Okay. And then I love the fact that you bring up the bone marrow derived macrophage, and yeah, there's that's one of the reasons why that there's gotta be M1 and M2 there, even with the tissue resident. So you know that's probably why it's so in those three tumor types that we see more. And I you know definitely like that observation. Bone marrow derived, and then the role of IPI, I wanted to just share this, and for the last, I know you don't have time, but from the DART study, the Nivo IPI study, now we're working with CMAC group, and their first lab is in MD Anderson, so we're working with, I don't know if you do know Kara Haymaker.
Yeah.
She's the CMAC PI that I'm working with. And what we found is in neuroendocrine tumor, we saw pretty good response rate, like 40%. And we're looking at the tumor microenvironment with NanoString, and what Kara showed us in her lab is that with all our trial samples, that she found the T cell density does not predict. It's the B cell and the tertiary lymphoid structure. And probably that is what's bringing in T cells. You need T cell, but T cell wasn't there in the first place. It and the PD-L1 level, TMB, nothing correlates, but it's the B cell. I mean, we didn't have protein marker yet, but this is all NanoString based. So B cell signature and macrophage signature, dendritic cell are the ones that predicted response. So it's not just an Nivo, but it's probably IPI bringing in, and B cell, you know, it's still in its infancy. We, you know, there's T cell, Treg, and then there's a B cell and Breg, and people are studying, you know, they don't really have good clear biomarker for those. But we're really thinking, you know, B cell, and the B cells usually at the tertiary lymphoid structure. So I don't know, Dr. Taegon Kim, do you have any thoughts on the role of, I know you're doing liquid malignancy, but more like you studied melanoma and other things before. So the role of TLS and B cell, what is your thought on this topic?
So unfortunately, my background is a T cell, and then you probably understand I'm a little bit biased opinion about B cell.
Mm,
B cell may be one of the cell subset to help,
mm,
to meaning more antigen presentation role
mm,
or dendritic cell as well, and then help some generating more strong effector T cell.
mm,
But I am a strong believer without the antigen specific T cell,
mm,
there's no immunotherapy effect.
mm,
So let's say somebody, macrophages, we're going to stimulate the macrophage.
mm,
Yes, initially, yes, the macrophage is doing something,
mm,
but the final effector are basically T cell.
mm,
So without T cell, there's no immunologic anti-tumor effect. That's my personal opinion.
mm,
But unfortunately, this is beyond my expertise, like B cell.
mm,
I know that many people talk about regulatory B cell, and then some of the role of the antibody also important. Yes, because the tumor microenvironment is very dynamic, and so we don't know what is the main player.
mm,
However, I still believe that can help and then adjunct to T cell based anti-tumor effect. So I'm not sure, based on my knowledge, I don't know how the CTLA-4 bring the B cell. To be honest, I don't know.
Oh, it doesn't bring B cell. B cell was there before. And then
Okay.
if you use EP, then those with B cell, then if you add EP, then EP brings T cell to the tumor. So that's a new discovery we saw with our DART patients. So we're trying to make sense what's happening.
Yeah, so interesting. But so my mentor Liping didn't get along in the Jim Allison, and then the one day he asked me to review the manuscript called the reprisal of the ipilimumab, and that's basically ipilimumab is strong dominant effect of ipilimumab is basically the deplete the Treg. And then after the Treg depletion, then add some Nivo, then we don't know that actually augment the anti-tumor effect. I don't know. But the main issue about that combination, I have 5 minutes, but I need so EP plus Nivo, especially EP plus Nivo. So I don't think overall response is going to be our endpoint. Basically, overall survival should be our endpoint, because and then toxicity. Because initial melanoma Nivo EP trial, we see the great response, but as good as the Nivo alone, but the toxicity is higher, and then eventually overall survival no difference, or sometimes worse. So that is the reason I have some feeling at the end of the day, in 2 years, FDA reviewed the ipi-nivo combination in the hepatocellular carcinoma, and then they, I don't know how they want to comment about that, but BMS push a lot this combination, yeah, as you know.
Yes, they do. Yeah. Well, at lung cancer, it looks like it's durable compared to Pembro or Pembro plus chemo. But I think melanoma and lung are also very different.
Yeah.
Okay. Well, I don't want to, take too much of your time. I want to, acknowledge your mentorship and input during our that meeting, but we are probably gonna invite you again when we
Oh, yeah, thank
Yeah.
you, thank you. That's
Yeah.
fun. And then maybe I can invite you and Minjung to others and to our lab meeting, and then you guys are surprised that some of the member cry and
Yeah, I will definitely invite you when Daniel Shin joins. We can also talk about interferon gamma. Yeah.
yeah, don't make him up too early. Maybe it's a seven or yeah, California time.
All right. Well, we'll let you go and we'll proceed with other members.
All right.
Thank
Very
you.
nice meeting you. And then maybe I will visit Chicago in January next year, but my daughter's fencing tournament again. I will reach out to you, and then hopefully, if many people are available, hopefully I can have a dinner with you guys.
Yeah, you can come to our lab. Yes. Thank you.
Okay, bye bye.
Thank you.
Thank you.
Yeah, bye bye.
All right, very good. Okay, Minjung, while you're on, do you have any updates or comments or questions or anything? I know you're in Korean time, so.
No questions for now. I think I'll have to do a lot more reading.
No, I want to emphasize, remember what he said? He used the word beautiful. So this is the kind of intellectual curiosity and stimulation you need to be in this field. Anytime you see data and interesting phenomena, it has to come as a beauty. Whenever I meet with Dr. Taekwon Kim, we always talk about science, and it's just like we talk to each other. Like, we're so happy that we can talk about science, the new things. So there's that joy and excitement. I think it's really good that we have Taekwon Kim as our mentor. Number two, I want to emphasize, the expert or guru, whatever you want to call it, they know what we don't know. So that's how you can call them expert. Like, we don't know why, no one explained it better. So, getting this perspective is good, because if you read some review article, you feel like they know everything and they can explain everything, but they don't know. So the experts know what the field doesn't know, what they don't know. So people who act like they don't know, they know everything is imposter. You cannot trust them. I just want to say it for you guys, that it's very important that you know what you don't know. So yeah, so look into the PD-1, PD-L1 and find more articles about M1, M2, and then the association about the EGFR. Maybe in a week or two, maybe you can give us update on what Dr. Taekwon Kim told us to look at, and then on that, and then do another separate journal club. And by that time, let's see if we can invite Dr. Daniel Shin and maybe give him like 8 a.m. California time. It'll be 10 p.m. But if you can still stay awake till like the midnight, I think Dr. Daniel Shin is the leader in the field. He's been invited by the Korean Cancer Society to give plenary speech. His mentor, Dr. Tony Ribas, is the leader in the field. So yeah, we could definitely get his perspective. So yeah, let's plan then in the upcoming next couple of weeks, Minjung. And where is your hyperprogression paper?
I think I actually have been delaying sending the pre-submission inquiries because while checking on some of the feedback that I received from JAMA during our first time, there were certain things that I wanted to kind of get clear before I do the submission again. And that has led to just a lot of restudying the statistical portion of the meta-analysis. But I'm finally wrapping up. So I think this week I'll send the pre-submission inquiries, and I'll have a better update next time on how things go.
Okay, so anything you updated, just present at our next meeting and let us know how you're proceeding.
Mhm.
Okay, very good. Good job. Good job. So yeah, I'm glad we are a team delving into these good questions. And the field doesn't know. I mean, that, our meeting with TK Kim, proved that not many people know what's really going on. And even people like Jim Ellison and Li-Ping Chen, they're the leaders in the field. We thought Li-Ping Chen was gonna get Nobel Prize, but he did, so he's pissed off. And Jim Ellison and Honjo got the Nobel Prize. So, for a field to grow, there are scientists who contributed in making the field grow, new discoveries, but only few, one or two get Nobel Prize. And there could be a political and, things going on. And so these people sometimes don't agree on things. Even with the scientific, evidence of, it's a matter of how you interpret the data. But it's good to know their perspective. Okay, very good. Thank you, Minjung. Okay. Maybe Yongjun Chap, I mean, it's good to have you. Do you by chance explain to us that what's your field of research, and give us like an overview of what you're doing, so maybe we understand what you're doing and see if there's any point of collaboration between your lab and our lab.
Yes, sure. But first of all, I want to ask you how much time is usually
In the lab meeting?
Yeah,
We
yes, for
usually do about like 3 hours.
3 hours. Okay.
8 to 11.
Okay.
Usually, yeah.
Okay, then I'll just briefly review what I'm doing now. Yeah. Okay. And I share my slide.
Oh, by the way,
Yeah.
Dr. Jian Lee, she joined late. Dr. Jian Lee, can you introduce yourself?
Hello, my name is Jian Lee. I'm working in Gyeongbong National University Hospital, and I'm a breast surgeon. Now currently working with Dr. Chen for research year. Nice to meet you.
Yeah, so she's
Yeah.
with us for 1 year. She's just like you, doing 1 year of research.
Yeah. Okay, so I just briefly overview what I'm doing. I'm Yongjun Cha. I'm a medical oncologist, and I treat mostly patients with colorectal cancer and some patients with gynecological and other gastrointestinal malignancy. And the contents I prepared is 3 studies I'm doing, and one is ctDNA-based adjuvant trial now ongoing, and the other 2 is some genomic research. Actually, I forgot I was supposed to present in English, so please understand. Although my English is quite bad.
Okay.
Yeah. So this actually this is a platform study. Now I'm co-PI with Dr. Seon Han of Seoul National University, and this is ongoing since early mid this year, and the name of the study is platform study circulating tumor DNA directed adjuvant chemotherapy in colon cancer, and actually is cases to support this trial, and comprised of a part 1 observational study and part 2 randomized controlled trial. And as you know, stage 3 colon cancer, the benefit of adjuvant chemotherapy is only limited to 25% of patients, and 50 patients are cured with resection alone. So we think that the accurate quantification of minimal residual disease could benefit patients in all these categories of this patient. So there are many MRD study with the ctDNA assay, and they show that MRD positive group has a very poor prognosis. And so based on this scheme, there are many ongoing clinical trials, and there are two schemes. One is adjuvant escalation trial, and the other one is the adjuvant de-escalation trial. And in some study, as in NRGGI 008, this combines the adjuvant escalation in ctDNA positive group, and also adjuvant de-escalation study in ctDNA negative group. As I think you already know the result of this study. It was published in NEJM this year, and it is a randomized phase 2 study dynamic, and it compares the strategy of ctDNA-based adjuvant chemotherapy in stage 2 colon cancer, and it showed that with the ctDNA-based adjuvant chemotherapy, you can reduce the chemotherapy, the proportion of patients who received chemotherapy, and when the survival of the both group is comparable. Yeah. So in this study, we are using some different type of tumor-informed assay called alpha liquid detect. It is composed of tumor-informed assay based on tumor whole exome sequencing, and also it analyzed tumor agnostic clinically actionable variant, very common in colorectal cancer. And in this ESMO meeting, the preliminary study with this alpha liquid assay was presented, and as you can see, when ctDNA MRD was analyzed at post-op 3 weeks, the prognosis of the patient is very different according to the MRD positivity. So actually at first we designed this kind of this scheme of study. So we in the part 1 of observational study, we enroll 1,200 stage 2 and 3 colon cancer studying adjuvant chemotherapy, and then we give the standard FOLFOX or CAPOX for 3 months. Then, if in case of MRD positive during the 3 months of adjuvant chemotherapy, then we intensify this patient with and FOLFIRINOX with 3 months additional FOLFIRI chemotherapy. So we saw that with this approach, we could improve this very poor prognosis patient. But in the preliminary study result, the MRD persistent group during chemotherapy was not that prevalent as we assumed at first. So we changes the design of part 2 study, and so the part 1 study is same. 1,200 patients are enrolled, and they are observed during over 3 years for their ctDNA MRD status, and they receive 3 months of FOLFOX or CAPOX treatment, and at the 3 months of period, they are if their post-op MRD status was positive, they are randomized into 3 months additional treatment with FOLFOX or chemotherapy. That is the standard chemotherapy versus and modified FOLFIRINOX of 3 months. So this is the overview of this platform study. And because of this is platform study, you can begin another interventional study during the observation study. And the part 1 study, the observation studies endpoint is the enrollment rate for part 2 clinical trials, and secondary endpoint is ctDNA positivity and ctDNA clearance rate at each time point. And this is the scheme of the part 2 randomized control trial. And the number of patients is 236 patients, and primary endpoint is 3-year disease-free survival rate. And the hazard ratio we assumed is 0.64. This is the outcome assumptions for stage 3 colon cancer. And with the patient with post-op MRD positive group, we assumed their 3-year disease-free survival would be around this 30% with standard chemotherapy, and we assume that their 3-year disease-free survival might increase, could increase up to 46% with the addition of immunotherapy treatment. So this is the overview of our study. Yeah. So actually, the part 1 study is now ongoing, and we are recruiting patients with stage 2 and 3 patients. Yeah. And yeah.
Can you go? I just have a question.
Yeah.
So the part
Yeah.
one more,
Yeah.
one before.
Yeah.
So, what was the decision about the 3 months of FOLFOX? Because some, like you said, are cured without even the 3 months. Did you guys discuss that? Because everyone gets 3 months of FOLFOX, K-pox, right? In this study, there's no.
Yeah.
Like, what if they're MRD negative right after surgery?
Yeah, so for MRD negative patient, actually, based on the recent IDEA trial, the non-inferiority of 3 months of K-pox is suggested. So you can just stop chemotherapy at physician's discretion. So in patient with a post-op MRD negative group, then you can just add 3 months of FOLFOX chemotherapy to complete 6 months of adjuvant treatment, or you can just stop treatment.
So you can do either one, okay? But what about between part 1 and 2, that everyone has to get FOLFOX K-pox?
Yeah, because we enroll patients with high risk stage 2 and stage 3, and the standard treatment, adjuvant treatment for this patient is 3 to 6 months of FOLFOX or K-pox. Yeah.
So this is not a de-escalation study.
Yeah, this is an escalation study.
Escalation study.
Study for patients with post-op MRD positive who is very high recurrence rate.
Are you planning to do any de-escalation study?
Actually, we planned the de-escalation scheme, but it requires more patients. Yes, the size of study should be bigger, And we have a very limited budget, only very limited budget. So this is the main reason we chose the escalation study. Yeah,
Yeah, we
Yeah.
thought about the same thing in lung cancer.
yeah,
Escalation, it required many more patients.
right,
So
right, right,
I share your,
right.
yeah.
Yeah, and one other concern is that many oncologists in Korea are concerned about not giving chemotherapy
Mm.
when the giving chemotherapy is now standard. So
Yeah,
then yeah, so
ethical concern, yeah.
yeah.
But are you aware of Japan Circulate, the
Yeah,
Natera data?
yeah, sure, yeah.
It's the de-escalation study, right?
They are actually combine escalation and de-escalation scheme.
Mm.
There are observational study and one escalation and one de-escalation study.
Yeah, but their de-escalation study showed non-inferiority, right?
Yeah,
Yeah,
right.
I think they had a larger sample,
Yeah.
but it
Yeah.
it is same stage 2 and 3, right?
Right.
Yeah,
Yeah,
and they omitted any FOLFOX for the MRD negative right after surgery.
right.
Yeah.
But actually, their escalation scheme is
Different.
somewhat weird from our usual perspective because for in their study they give just fluorimidine monotherapy.
Oh,
Yeah, for MRD positive groups. So I understand their decision because they need support from pharmaceutical,
mm,
but actually that is not now sound from our perspective.
mm,
The design of study. Yeah.
mm. Okay, let's look
Yeah.
here, your second portion.
Yeah. So
One question. What
Yeah.
company ctDNA are you using?
is the IMBDX?
I've not heard this company.
Yeah. Actually, it is a startup.
Startup?
The startup, and the founding member is, Dr. Tae-yu Kim of Seoul National University
Yeah.
Hospital.
Yeah.
Yes, and he and Professor Do Hee Bang of Yonsei University Department of Chemistry.
Mm.
Yes, he is a co-founder of the company, and yeah.
And then they do both informed and agnostic, both.
Their panel is alpha leak detect, and it is more likely tumor informed assay. They are tumor informed assay,
Uh-huh.
but contrary to other tumor informed assays such as Natera,
Yeah.
they also include some clinically actionable variant in addition to tumor, the patient-specific variant found from tumor whole exome sequencing. So it is some difference. And with this approach, you can know the new variant at the time of the MRD positivity, MRD positive conversion. So it is one advantage for that approach.
Is this clear certified in the US too?
No, it is just the result is just published in ESMO, and it's now is preparing for submission.
Oh, okay.
Yeah.
Okay.
Only very preliminary, but it is this very same situation with Natera and other Guardant. Yeah.
Got it.
Yeah.
Thank you.
Yeah.
Okay, you can move on. Thank you.
Yeah. And the second study is whole genome and transcriptome sequencing of oligometastatic colorectal cancer. And as a colorectal cancer specialist, I have I'm always curious about the phenomenon of oligometastasis. And as you all know, in colorectal cancer, liver metastasis or lung metastasis, even though when you have distant metastasis, some patients, around 20 to 30% of patients, they can be cured with resection alone without chemotherapy. So
Mm.
it is very unique phenomenon. And also the concept of oligometastasis is now being widened to other indications, as I know. So it is very unique phenomenon. And so we think that for metastatic colorectal cancer, most patients have a widespread metastasis and they can be controlled with chemotherapy, but some of the patients can be curable with resection alone, and some can be curable with resection plus chemotherapy. So this is a phenotypic spectrum of metastatic colorectal cancer. So the study objective is why some tumors are curable in the presence of cystic metastasis, and how to identify and treat curable subset. So it is the study question, and the study objective was actually it is a pilot study, feasibility of a whole genome and RNA-seq analysis, and also genomic and physical features associated with long-term survival. So we have prospectively maintained the colorectal liver metastasis registry, and we have around more than 600 patients in the biobank. And so from those patients, we chose 20 patients with treatment naive before treatment sampling, and with more than 7 years of follow-up, and also synchronous colorectal liver metastasis. So we analyzed the primary and liver metastasis tumor and also PBMC sequencing. So this is basically characteristics of 19 patients. One was excluded because of very low tumor purity. So mostly all patients were MSS, and most patients received the post-op chemotherapy without neoadjuvant chemotherapy. And one particular strength of our study is that most patients have been followed very for a long time. So they more than 6 years follow-up, and around the 30%, around the 40% of patients remained disease-free more than 10 years after 1 or 2 resection. So you can, so the progress of the patient is divided binary, the cure and death. So it is, I think it is a very unique setting. And this is oncoprint, and major pathogenic variants found were consistent with TCGA data, and also they are conserved in liver metastasis. And similar result with the SMV, most are shared, and 20% were liver metastasis specific. And tumor mutation burden was not actually associated with the recurrence of survival. And when we analyze the mutation signature, interestingly, SBS signature 3, that is of defective HR DNA repair, was significantly enriched in this survival group. But actually, honestly, we don't know the explanation for this phenomenon, and I'm not sure it can be reproducible, but in a way it is hypothesis generating, and I think it's an interesting finding. And the copy number variation, so we analyzed this structural variation with tumors with tumor cell content more than 20%, and most recurrent most CMV was observed in recurrent event that is already observed in TCGA. And one interesting finding was that chromatrix event was found in 3 cases, and all was observed in TP53 mutant group, as you can as you can think. And this is the phylogenetic tree for this patient.
Mhm.
And we have 5 patients with this free group and 11 patients who recovered and succumbed to death. And the 2 patients in the recovered group and 1 patient with this free group have a chromotripsis event revealed by a high confidence score. And interestingly, in this free group, the chromotripsis event was only observed in liver metastasis, that is private in liver metastasis, not present in the primary tumor. And contrary to that, in record group, the chromotripsis event and structural variation was shared between a primary tumor and liver metastasis. And it suggests that in this very poor prognostic group, the evolution of tumor metastasis was very rapid at the first and also the evolutionary bottleneck for their metastatic spread is very wide for this patient. So I think it's a very interesting finding. And we next analyzed transcriptomics, including CMS, and is consistent with the previous publication. And also in the record group, we found that ER pathway and TGF beta signaling pathway was enriched. And in the immune cell fraction analysis, actually it is bulk sequencing, and it is just it is performed the tumor bulk, so it is not accurate.
What algorithm did you use? Excel, CyberSort?
CyberSort, yes, CyberSort, yes, CyberSort. So, so interestingly, Treg and macrophage M1 in primary tumor with associated with better survival, and macrophage M2 in primary tumor and also in the liver metastasis correlated very worse survival. So it was very interesting. And so in conclusion, we in this pilot study was it is although very small study, we think that the setting of the study is very unique in that we include patients with a very long survival and binary outcome. And we found that the immune cell signature and also early genomic event showed by primary tumor liver metastasis might explain this recurrence in post survival in oligometastatic tumor. So, so we are now preparing submission of this study, and the next study is actually I just received the result of the study a couple of weeks ago, so I have not looked into in depth, but I want to show you the overview of the study and the preliminary result. In this study, actually we performed multi-regional whole genome sequencing in 7 patients. Although the number of patients is very small, the unique point of this study is that we analyzed in spatial and temporal samples from these 7 patients. It means that we collected a primary tumor and also their paired liver metastasis, and also the other metastasis that is more than months or years later.
Mm.
And also the second strength of the study is that we performed the single cell sequencing, single cell whole genome sequencing. So actually, the number of patients is only 7. The number of whole genome sequencing of this study was around 100. So, so some of the primary tumor, we analyzed the whole genome sequencing in organoid, clonal organoid, in addition to primary tumor, and also metastasis and second metastasis. So the reason why we performed the single clonal organoid sequencing is that, as you know, we want to actually analyze the single cell level genomics rather than the bulk. Then when we analyze the whole genome at the single cell level, we can we can see the accurate portrait of the genome. So we want to analyze the whole genome at the single cell level. But for single cell analysis, you need many cells. So you have to pick organoid, and then you need expansion. Then after single cell sorting, then another expansion. So and then you perform whole genome sequencing. So you can analyze around 5 to 10 single cell whole genome sequencing from one tumor. So this is the total mutation and mutation signature composition in each patient, and this is driver mutation heat map. And this is one example of the patient. This patient received primary resection of sigmoid colon and was analyzed with whole genome sequencing. And then he received FOLFOX chemotherapy. And then after 1 year, he received peritoneal seeding was found and resected and was analyzed whole genome. And then he received bevacizumab and FOLFIRI and FOLFOX chemotherapy. And then at the time of another peritoneal seeding progression, then his tumor was collected, and whole genome was analyzed in his peritoneal seeding primary tumor, and also in the clonal organoid. So in this approach, we could find the mutation signature very accurately. When we analyzed the bulk sequencing only, many mutation signatures are not revealed with bulk sequencing, but with the single cell sequencing, you can accurately describe the mutation signature, and also you can analyze the genomics of each tumor accurately. And yeah, and this is another example. And actually, we are not, there are only 7 patients, and the findings from this study is very complicated. And so we are now drawing some common theme from this result. But the most interesting finding for me was that we could find many convergent evolution of driver mutation. Even though it is at first I thought that driver mutation such as NRAS, KRAS, BRAF, or HER2 mutation, HER2 amplification would be very conserved from the initial tumor to the metastatic tumor, but it was not that likely. And even though it is a very strong driver, the mutation was not conserved, but it might be plausible because colorectal cancer is well known for its chromosomal instability. So in this cancer with chromosomal instability, the heterogeneity in driver mutation is very frequent, and also even though some driver mutation was found, actually it can be found in some other chromosome. 그러니까 for example, you can see big amplification in many primary tumor in primary tumor, and also their paired metastatic tumor, but the number of amplification was quite different. The level of amplification was quite different, and for patient with HER2 amplification, you see HER2 amplification in primary tumor and their metastatic pair, but the site of amplification was different. In primary tumor and metastatic tumor, the different loci, the different allele was amplified. So this kind of convergent evolution was quite prevalent in this in our study population. So I think that this kind of convergent evolution of driver mutation and their heterogeneity is very interesting for me. And so we are now reviewing and wrapping up what we found, and we are summarizing now, and it's very preliminary, but I wanted to share with you. That was the end of my introduction.
Wow, this is really deep genomics transcriptomics. Thank you for sharing. Maybe for our lab members, can you explain the concept of convergent evolution, just more like a concept so that they understand?
Yeah. When for example, when we think of KRAS mutation, you commonly think of all tumors in the same patient have same point mutation in KRAS, but actually it is not. Some patient in patient with KRAS mutation, some might have some different mutation in KRAS loci, and also in the HER2 amplification, you can observe some different region, the amplification in different region, and also the level of amplification between tumors. So it is called convergent mutation or convergent evolution, and I think it is very interesting. And I think that can partly explain the relatively lower response to targeted therapy for colorectal cancer, even though they have the same HER2 amplification or BRAF mutation.
Thank you. Yeah, I've seen similar studies in melanoma and other cancers. I haven't seen something like this in colorectal. So yeah, I like dissecting all the phylogenetic trees. And what was the clonal pressure that each therapy had. It will be interesting to see what's going on with MSI high and Nivo-IP and
right.
But and then looking at your presentation, I've treated a few colorectal patients. If they're Korean patients, I still treat. I usually treat more thoracic, but some people are Korean speaking and they want me. And then the chemo is very standard. It's not. But then I've noticed that in other tumor types, it's hard to recycle chemo.
Yeah.
But in colorectal, you can recycle. Like go back to bulk and it works again.
Yeah.
And I don't know why it's unique to colorectal. I don't think other tumor types I've not tried or seen it much.
There are two reasons.
Yeah.
The one reason is that there are very limited number of effective chemotherapy agents in colorectal cancer, so you need to reuse it.
Yeah.
And also the other reason is that in colorectal cancer, the progression-free survival with FOLFOX or FOLFIRI-based chemotherapy is relatively longer than those found in stomach cancer or other gastrointestinal malignancy. And the toxicity with chemotherapy, for example, oxaliplatin-based neuropathy, that makes us to stop chemotherapy even though they are still responding. So you need to stop, and then you can have a treatment-free interval. So we should recycle them. Yeah, that is the two reasons. Yeah.
Yes.
Yeah. Actually, I don't like reusing chemotherapy. I want to use immunotherapy or other targeted therapy, but the option is very limited. And
Right.
the progress in the development of effective chemotherapy is very limited for field of colorectal cancer. Yeah. So,
Yeah. And neuropathy with oxali is real. Some
yeah,
people
right,
never comes
right.
back. So
right.
my mentor was Scott Kopes and MD
Okay.
Anderson.
Yeah.
I was in his clinic and
Yeah.
he always start with irinotecan because
Yeah.
he doesn't like oxali. So
Yeah.
he doesn't start with FOLFOX.
Yeah.
But anyway, yeah, there's always good questions. But yeah, I think everything you're doing, we like to do that we're definitely interested in lung and breast and other, I think we're kind of doing the same thing across different tumor types, and
Yeah.
it's good to see that you're doing that in colorectal. And the local therapy cure, I saw the JAMA surgery paper, liver transplant for patients
Right.
liver,
Recently. Yeah.
just the liver mets, and the colorectal
Yeah.
primary was resected, nothing
Right.
there.
Right.
So Dr. Jian Kim also is looking at the data for lung transfer plan for bilateral lung cancer. So
Yeah.
that's what we're pioneering here at Northwestern.
Yeah.
It's stage 4, but if you transplant it, if it's
Yeah.
not going outside lymphatics bloodstream,
Yeah.
so I think whatever you learn, like what makes it not, metastasize through lymphatic and
Yeah.
bloodstream and just stay in liver.
Yeah.
What is in the same for some brain metastasis with lung cancer are cured.
Yeah.
Same thing.
Yeah.
So what is the unique biology behind it? And in lung
Yeah.
cancer, we're looking at spread through just the airway,
Yeah.
not through lymphatics or bloodstream. And we're
Yeah.
trying to understand that. So I feel some overlap in that question as well.
Yeah.
Very interesting. Well, thank you for your time, and I think whatever you learn, we definitely can from your whatever you learned, we can learn from your research. I know you're busy, but you are welcome to stay till the end of our lab meeting.
Yeah.
People are doing in our lab.
Yes. Thank you.
Um,
I'm happy here.
yeah. And then you are always welcome. So it's always this time, 8 to 11,
Okay. Yeah.
every Tuesday. So
Yeah.
you're always welcome to come and discuss together and learn. Yeah. So why don't we start? Well, anybody has any comments or questions for Dr. Cha? No? Yeah. So these are the talks that you'll hear from KSMO or AACR or
Yeah.
ESMO. but we heard it in our lab meeting, so it's an honor to have him.
Thank you.
Dr. Oh, you have a question?
Thank you. I mean, I just want to comment and then it may be a silly question, but if time allows, I really want to ask you a little bit of question about the single cell analysis.
Yeah.
Also, so like, thank you for letting us hear about your researches, and it was very fascinating. And just seeing that kind of phylogenetic degrees,
Right.
it was very new to me. But I think it has a great potential to reveal a lot of information about the cancer.
Right.
But like, so at this moment, what I'd like to know more about the applicability of that technology. Like, is it, can we use that to actually find the next treatment options for patients, or like, what's kind of a driving mutation for resistance? Like, what's the purpose of that, practical purpose of that technology at this moment?
You mean single cell sequencing? But, yeah, but yeah, I think that it is very early to talk about single cell sequencing in clinical application. Actually, we even for bulk whole genome sequencing, it is very early yeah, in the clinical application, and yeah, but because but we can analyze more accurately the genomic portrait of primary and metastatic tumor, and also the mechanism and course of metastatic spread with the single cell sequencing. So I think that it is very early to apply the single cell sequencing into clinical practice, but it has very much potential in research, especially for using the tumor dynamics in the treatment sequence, and you can determine the sequence of local and systemic treatment, and also duration of treatment. When you can assess the tumor dynamics and the process of metastatic spread. So in that point, I think that it has many potential in future, but not now.
Yeah, couldn't agree with you more. Yeah,
Yeah.
thank you for answering.
Great. Thank you, Dr. Cha. I think we'll start with Dr. Eugene Oh, because she's in Korean time. Do you have any updates, Dr. Eugene Oh?
So I'd like to firstly update you guys on the hyperprogression project, which we discussed a lot earlier. Thanks to Dr. Jiyeon and Jisang, we were able to get the progression data, clinical progression data on each regimen, and I finalized the table and the results. So if time allows, could you briefly confirm whether we could use this table as our final material? So
yeah, you have it. So
yeah.
I noticed that we were kind of doing this nonstop for 2 hours. Let's take a five bathroom break, 5 minutes,
Yeah.
and then we'll come back. Okay, 5 minutes. Thank you.
Mentioned in the table and script, maybe table 2 can be a supplementary table.
Yeah, I saw that
Because
comment.
it's not much important in this this
Yes.
manuscript. So
Right, because we decided not
yes.
focusing on the
Mhm,
new definition. So
mhm.
Yeah.
And there are some journals which mentioned about the number of limitation about the tables. For example, analysis of surgical oncology is limitation is only 5, including figure and table. Now it expanded at 7, but if we span the 5 tables, then we can use only 2 figures for this
Okay.
journal when we're submitting. So
Right. That's so true. And
yes. And do we have a specific figures, Arnie?
figures, we do have like survivor figures, you know, PFS and OS.
Yeah,
I
it
think
could be
I
2,
think I
right?
think I think
I will
pictures.
make it into one.
Eugene, do
for
you
the-
remember there is one time that Dr. Che show us like a plot chart, or I don't know, I think for the hyperprogression one that only that we choose. He once showing like this is like the real hyperprogression, like the like the XY chart, but like this kind of chart, but it's like have like from this and to this. I don't know what to say. I call this like a plot chart, but it's showing like
Oh,
the,
yes, we have one, we have to share the figure for
what is
flowchart.
that? What is that figures? I forget the name of the figures. I don't the one like this and and showing like
Dr. Che, we're
sorry.
already discussing. Sorry.
Let's resume. Okay, Eugene, show us the figures.
Of course, just give me a sec. Could you see the screen? Okay, so originally, yeah, but you know, like me and Dr. Jian talked about whether there would be too many tables to be contained in just one article. So she gave me advice that, you know, we will keep the table 1, just basic table characteristics and factors for hyperprogression, kind of demographic table, and then we have this kind of table 2 comparing the definition and the modification we had made before, but because we decided not too much focusing on this kind of concept, so as she recommended, I think it's good to move this table to a supplementary portion section. And table 3 is
It just
also
where the resolution of your screen is not good. I can't read anything. So
okay, then
everyone like read.
oh, I didn't know about that. Let me just reshare if that's not too much.
Was it just or did you guys able to read the screen, or it was just very bad?
Yeah, same here. The resolution is quite
Okay,
poor.
got it,
yeah,
got it.
yeah.
Yeah, maybe. Yeah, I just bought another separate screen, and I think it doesn't have that much good resolution. And here,
So you, Dr. Cha, this is a project on what are the predictors of hyperprogression, and how does hyperprogression affect overall survival?
Mm. So it is a meta-analysis,
no, it's patient data retrospective
a
analysis.
retrospective data.
Yeah,
Mhm.
just our own Northwestern data analysis.
Yeah.
Yes.
Mm.
So
Yeah, it's better. We can.
see, yeah, get better. So table 1, patient characteristics and associated factors of hyperprogression in lung cancer. So Dr. Chai, we included in total 128 patient data, and
Well, it
yeah,
blurred again.
I think
blurred
it,
again?
Blurred.
maybe
Yeah.
move it just happens.
I'm
Why don't you
sorry
open
about.
in Word or something
Oh, not in
other?
Google. Okay. Yeah, I'm sorry for taking up too much time just to. Okay, let me just try to download it as micro Word, and Hopefully it'll be better.
I think it's the whole screen, not, I don't know why this is happening.
If someone else can go before me,
Or if
I
you
would-
have a link to Dr. Jian, maybe Dr. Jian can scroll it for you.
share. Of course, sure. I
Well, now
didn't
it's,
I
oh, I guess as soon as you move your cursor, this happens.
Okay, let me share with Dr. Zion.
You sent the message in early morning, so I have it. I can
Oh.
share.
Yeah.
I'll just unshare my screen.
Please. Okay. Is this right?
I think, yeah, correct.
So table 2.
Table 2. Are we good to move it to supplementary table? Because the main idea of this table is to compare definition considering only target lesion and the definition that we made modification to include new detected lesions. So
That's fine.
okay. So we will move it to supplementary, right?
Okay.
In table 3, it just shows us the prevalence of hyperprogression defined by the different definition, Champia, Sarabuzit, and Ferrara.
Mhm.
But after this table, we only focus on 2 definitions because as you see here, Ferrara detects none of them as hyperprogression, having hyperprogression. So
Okay.
and could you scroll down? So this is the risk factors analysis result to find whether what kind of clinical pathological factors are associated with hyperprogression. And yeah, there are no changes made compared to the previous version of this table. So you could just scroll down. The main change was made in table 5. I'm sorry, like I'm still working on the title of the table. You can just ignore it as of now. But then I added progression-free survival results. So here, I used Cox hazard ratio analysis because it was time to event analysis. And so could you scroll down a bit? So the histology and smoking status, surprisingly, and then gender also all were correlated with progression-free survival. And with a marginal statistical significance, but still, and it all makes sense to me. I think squamous cell and small cell progressed a lot compared to the adenocarcinoma and others. And could you scroll down a bit?
Okay, suggestions. For those that are greater than 0.05 p-value, I wouldn't bold it. Okay.
Oh, okay.
I mean, it's a trend, but I wouldn't bold, make it bold. And for the second column, I would put the number of patients in like adenocarcinoma. What was the N equal and then percentage of that, and comma, and then percentage in the group. Yeah, and then that goes all for the all groups in the second column for other variables. So I think
Okay.
reviewers would want to see it. Yeah.
Do you want to add one more column or just put it in this column?
Just put it in there, it's fine.
Okay, all right.
Now, stop right there. So when Lee first presented the risk factors, he had PD-1 versus PD-L1. I don't know where it went, but I think just based on Dr. Taegung Kim's comment, we should add it as a variable. For only the monotherapy, within monotherapy, so I don't care if it's combined with chemo, it doesn't matter. But
Mhm.
in the single, so just leave it there, but create another column that says PD-L1 blockade versus PD-1 blockade just among the monotherapy group and see if there's any difference. I think Dr. Lee showed some difference in his first abstract, if you can find that. But anyway, with this new data, I want you to look at it.
Okay. Okay. So yeah, could you scroll down?
So I think small cell is controversial because small cell, if you don't treat or if it doesn't respond, it grows really fast. So I haven't seen like a small cell lung cancer study looking at hyperprogression. But I don't know if it's just a really small percentage. I don't know how many. What is your like guess about? Is it like 5%, 10%?
The percentage of small cell
Yeah.
proportion.
Yeah.
I'm expecting that would be in the table 1, and small cell is definitely yeah, 5%. Five patient.
Yes,
Patients. So less than
3.9%. It's only 4.
I see. And squamous is only 12.5%. That's odd. Huh. I wonder if you review others. I think it's, yeah, review it again to see if it's adeno or squamous. Sometimes it's just recorded as non-small cell lung cancer or poorly differentiated, but if you go to the path, you might see favor adeno or favor squamous. But yeah, I would review it again. So I'm thinking small cell lung cancer.
Okay, we're gonna review histology again.
Very different biology. So I would take it out. I would take it out from this paper.
Okay. Take it out means it should be included in others, or we can't exclude.
Yeah, I mean, if you exclude, then it's going to affect every
Yeah,
table.
sorry. Yeah,
Yeah,
we have to.
not just
do-
here. So how about it's you can add it in others then if you you can add it in others and see what happens. Yeah, because I don't want you to do the whole analysis.
Okay,
Yeah.
and we will review others again.
Yeah. Okay. Next.
Go back to this.
just to clear that, so added in others means like I have to remove this kind of classification in all kind of tables, right? So here not only in table 1, in table 4 and 5, I need to group small cell as others, right?
Right.
Okay.
Do we have more? Only table 4,
Table
1 and
4.
4.
4
I
and
think
5.
that's it. 4 and 5. Okay. Yes, you should.
Okay.
Mhm.
So we're looking at table 5, right?
Yes, this is
Okay.
table 5.
Could you go down?
Okay.
So not only gender or smoking history, yeah, you could go down. There are a few more factors that appear to be correlated with progression-free survival. So number of metastatic lesions and bone and liver. And I added hyperprogression as a factor to be considered when we analyze progression-free survival. So definitely it both of definition, Champion and Sarabujit, appear to increase risk of progression with strong statistical significance. And if you scroll down a little bit, so I included, do you think it's feasible to add progression? Here, progression means clinical progression that was collected by Dr. Jisang and Jian last week. And when I see the progression-free survival, of course, I didn't include it because it was a kind of result, but I consider this progression in over survival analysis, and it definitely was correlated with survival rate.
Survival, yeah.
So what is this progression? Is it clinical?
This is a clinical progression.
But
What
you're
you asked us to review.
right. But your endpoint is PFS, right?
Yes, so I didn't include a progression to the
You did
PFS, but yeah, so I made it as like blank. But I consider progression when I was analyzing overall survival.
But even for overall survival, I think it's redundant because it's the same time to event analysis.
Yes, yeah.
Yes,
And
I mean,
I have a
Carolina.
question. I have a question. For the progression-free survival and overall survival analysis, from what time point was it measured?
It was the initiation of immunotherapy.
Initiation of immunotherapy. Then
Yes.
I think that the event of hyperprogression is after the initiation of immunotherapy, and so it might be some strange to compare by the event according to event of hyperprogression. So because it is a time dependent variable, so I think that you can more appropriately analyze the post hyperprogression survival or something like that. Well, you can measure the impact of hyperprogression by some specialized analysis method, such as landmark analysis or something like that. And that's my comment here.
oh, thank you for your input, but yeah, this is honestly, this is the all things I am able to do as of now, but I would definitely learn how to do landmark analysis or something like that. But I coded hyperprogression as 0 and 1, and I just put that variable into the Cox analysis model. Would that be a problem? I mean, yeah.
So it can be a problem depending on what reviewers you meet. So it's almost like this. At the first scan, there's a progression. How would that correlate with the PFS? So it's it's the same thing. Like you have resist, but I guess if you're talking about the resist because we're talking about two different things. Where at first scan resist progression, and then your outcome is clinical progression. Because even if you have resist progression, if you think that there's a clinical utility, you can still continue. So we're defining clinical progression as time to the next therapy. Because your definition is different, I think you can still report it. I think it's a too clean marker. But I also agree with Dr. Cha's comment that it's not a true baseline predictor. It is the first scan. Based on the first scan, we're not even doing a follow-up scan. It's just like by the first scan, are we seeing hyperprogression or not? Would that predict clinical progression, which is defined by change in therapy? So it's a little different. If it's true RECIST-based PFS, and then you're looking at RECIST-based progression, you know you can't do it. So I would still leave it like this, take out the progression
Mhm.
for the purpose of this paper. But Dr. Chow, what do you mean by the landmark analysis? What are you proposing?
When you want to analyze the impact of hyperprogression on the survival, then you should analyze the only the impact at some time point, multiple time point during multiple time points. So
Like 6 months.
yes, 3 months, 6 months after you can analyze the impact of hyperprogression on the survival at 3 months, after 3 months, and according to the hyperprogression until 3 months, you can compare survival after 3 months, for example, and also likely for 6 months, 9 months. So it is some specialized method for the analyze the impact of time variable, time dependent variable. Yeah, time
So do
dependent.
you mean the baseline should be at the time when the hyperprogression is detected?
Yeah. So
But-
you can compare the survival only after the time after hyperprogression. I think, yeah.
First, like Dr. Che, isn't like the progressions. We want to find the progressions. The last table is the one that Jisung and Jiyeon find to checking whether this is like have a really clinical hyperprogressions or not, right? That's what we're checking. If we delete that table, so how do we know about the clinical hyperprogressions? That first, and second, I think I correct that the hyperprogression as the baseline, but how can we know it's hyperprogressions and we put it as a baseline if we didn't check it the baseline that we had right now? So do you think we still need to delete the last table? Because I think that's like the confirmations of the clinical hyperprogressions.
So I think that the true impact of hyperprogression on the outcome for survival endpoint can be measured with more appropriately with other kind of the survival analysis method. Maybe that's just my opinion. Yeah. Yeah. So because when you have prolonged observation time, then you have more likely to have a chance to observe some event, and that can be confounding. So I think that in this case of time dependent variable analysis, you might choose some different method. Yeah, it might be helpful, but I'm not sure.
Yeah. But I think still the baseline should be the initiation of the treatment. When we analyze the survivor after surgery, we analyze from the surgery and the at the point when the patient is still alive. That is the period. So for this study, because we are investigating the hyperprogression for the patient who received the immunotherapy. So I think the baseline could be the initiation of the immunotherapy.
So let's do this way. Hyperprogression. Hyperprogression is defined at the first scan on by the RECIST measurement and many like their definition. So we will know who will yes or no will be defined at the first scan, and everyone will likely get the first scan. So the yes or no, and then so because we're using different definition, although it is a time dependent variable, we have to wait till we get there, but it's a dynamic marker. So you could argue that the progression-free survival is going to be, the clinical PFS is the time the patient remains on the PD-1 inhibitor. So I think clinically you can still report it because the definition's different, but I also agree with Dr. Cha that landmark at, 3 months, 6 months, I don't know, maybe 1 year. Maybe something that we could do additionally to show the impact of, but even for that, I think to make it clean, doing it from the initiation of immunotherapy makes more sense. So we're doing scans usually at 6 to 9 weeks. So the 3-month survival will be, after the designation of the hyperprogression. So maybe just for hyperprogression, not for other variables, and you can, just for any clinical trials, you can draw HPD positive and negative and show how the 3-month, 6-month numbers change and what's the p-value between those points. I think you can do that to show the impact, and then you can do the exact same thing for the overall survival.
Okay,
So let's do
So-
that.
so 3 months and 6 months would be enough, or 12 months should, we have-
3, 6, 12.
Yeah,
about it.
3, 6, 12 should be good. Yeah.
And what we did investigated the clinical progression. I
Uh-huh.
think it could be included in table 1. It is just a clinical factor.
Mm.
We should remove it in table 5, but it can be included in table 1
Mm, 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 mm.
with the numbers of patients.
Yeah, Dr. Cha shared us this article.
thank you, I'll,
Yeah, later you can refer to the paper. It's it regards on the time dependent variable analysis. Yeah.
Yeah, Eugene, why don't you read this and reflect that in your analysis?
okay, I will.
Okay.
Glad to know that.
We will save it.
Okay, Eugene, anything else? Or what about, you showed us the overall survival before, right?
So
Yes, and then Dr. Jiyoung, could you,
okay, okay,
There-
okay.
things that I want to be confirmed? Yeah, and then,
I can't say.
so Dr. Cha, like, when we adapt that kind of new analysis plan for to see whether hyperprogression is correlated with overall or progression-free survival, can we still make adjustment in terms of considering other confounding factors to that kind of model?
Yes. Yeah. Yes, in the actually there are many analytical method, and for example, landmark analysis you can analyze multivariate.
I can do
Yeah,
still
yeah.
multivariate.
Yeah.
Because as of now, I did multivariate using just simple survival analysis, and I came up with the models,
Yeah.
but I think I cannot use that, and then try to get some new model with new analysis method.
Yeah.
So-
But actually, I'm not that is expert in the field of statistics, so you can consult with other statistician or yeah. That's the one.
Dr. Jiyoung, could you just go to the very end of
Okay.
the
Table 5.
even below table 5, there are some graphic.
Oh, okay. Oh, well.
Yes. So this was actually the model that I, selected based on the univariate analysis results. So, okay, then I will do this pretty similar thing with landmark analysis.
Mhm.
And, but I'd like to confirm that, okay, Dr. Che.
So you, Dr. O, you don't have this, so so you have this for univariate, and did you do it for multivariate?
Yes, I for the multivariate, I selected variables depending on the univariate analysis results as you
Yeah.
taught me. And yeah, as you see here, I think, yeah, so CPD is clinical progression. So in that model, I incorporated hyperprogression, which is dynamic variable. But I didn't have that when I did that. And then liver metastasis, the number of metastatic lesion, and MLR, and histology, and line of treatment, but when you like consider all the factors, it shows like only weirdly NLR and line of treatment were only marginally significantly correlated with clinical progression,
Mhm.
not even with the hyperprogression. I don't know whether it's correct way to see this result, but compared to this Champia, Sadabuzid was remaining as significantly
Mhm.
correlated factor even after adjusting for confounding factors.
Mhm.
So there was kind of difference.
Yeah, so multivariate can be done with the Cox regression like this. I think, again, you can't do it if it's you're using the same resist. We're doing it because we're only using the clinical definition of progression.
Yes.
So I think you can leave it. But for the landmark, you don't have to do the multivariate or univariate. You can just like the clinical trial report, you can draw two graphs, and at each landmark, you just show the progression rates, and then put p-value or the you can, statistically you can also draw the confidence interval as well, but
Okay.
yeah, just that would be, you don't have to do it for other factors or do multivariate for landmark.
Okay, got it. Could you scroll down? Because I do have results for overall survival as well. So yeah. Oh, I'm sorry about that. I think this is the results for progression, and then the above were about overall survival, because I included clinical progression as a confounding factor when I analyze overall survival data. But then I realized, I learned today that I need to remove that because it's kind of confusing.
Do you want to go back?
Yeah, so in the above model,
Above
I will
one?
delete
Okay.
I will delete
First one?
the yes. Why there are so many? So just my only question is, when I analyze overall survival, I cannot include progression as clinical progression as confounding factor, right? Is that correct?
Correct.
You can.
Yeah, I don't have any more questions about this.
Yeah, you
Mhm.
cannot, but you can put hyperprogression, yes or no.
Yes, hyperprogression, I will definitely include that. Yeah. Okay. Wow. So we need a few more modification on this
Okay.
data, and I will try my best to come up with a finalized thing by next week.
Yeah.
And
By
That's
next week, my expectation is Arnie finishes all the manuscript writing, okay? We don't wait for the result to come out. All the
Mhm.
writing should be done, introduction, discussion, it's all based on the previous reports. So whatever other,
Actually, the introductions already on making. I asked but I still like ask Jion and Eugene like to give me like feedback, and yeah, with right now just like doing the introductions. Maybe I will start with the discussion for next week, Dr. Che.
Yeah.
Yeah.
So work on it and get
Mhm,
feedback from each other. Don't wait for other people to give you a result. Write everything out, finish the manuscript so that it can come out all together. Because whatever Eugene will do, it's just modifying a little bit. We know the results already. So it's all about writing. You have to keep writing and reference and just help edit proofread it amongst yourselves so that when you present, I don't want to go over the English grammar. It has to be perfect. So maybe Liam can help with the grammars and everything, but so discussion, you know what to do. So discussion, this is writing class 101. Discussion number one, what is unique about our study that other studies have done? What is the first thing that we've done? What is the first new finding that's unique to our study? Has to be the first paragraph. And then the second paragraph is what has other studies showed before? What did we learn about the other studies? And then the next paragraph is about compared to what other studies have found, what did we find in contrast? They said this was correlated with that, A and B were correlated. In our study, we didn't see that. Rather, we show that these correlations were seen. So what's unique is other papers have not compared different definitions of hyperprogression, but we did. So by doing that, what did we learn? And what's on another thing? We talk about, hyperprogression, we see a lot of new lesions pop up. So can we propose a new definition, including the new lesion? We know the classic RECIST criteria does not include new lesions. It's just yes or no. But if you want to quantify, better quantify the hyperprogression, can we actually add the new lesions? And if we do, what do we actually see? So you kind of put our finding in the perspective of what the field has been doing. The field didn't do this. We're the first one to do this. If we do it, what are the new things we find? So these are the things you write in the the discussion, and the point is, and then what is the limitation? I mean, small sample size, and so retrospective and so forth, and and then what is the future? What do we need? What kind of study do we need? I would say I would emphasize the do no harm principle. Like actually, we still see that these people are getting hyperprogression, and we notice that they're doing worse. So how do we overcome that? Well, from our study, the immuno only is a risk factor. So if you add chemo, it's better. And that has been consistently seen throughout other studies. So you just back it up with other studies. And then the future is really trying to find clinical characteristics, pathological characteristics, or even radiomics characteristics of people who are going to get immunotherapy-induced hyperprogression, and larger studies, or and then we can cite our own studies like the UNOS abstract, and Dr. Yoon, I think Dr. Minjung Kim's meta-analysis should be presented at AACR, but any previous our work on it abstracts should all be cited, saying our group has shown that, and cite all the abstract that we presented on hyperprogression. And because just say our groups, this is the group story we're building together. So that's kind of the discussion in a nutshell. So Arnie, my expectation is that the writing finishes when the analysis finishes. Okay. And if you need help, talk to Liam, Dr. Jian, Eugene can all help you. The writing is a daunting task, but if without practice, you can't do it. Okay. All right. Anything else, Eugene?
Thank you,
Um,
Dr. Che.
Dr. J, and I will review the histology within this week.
Thank you. Thank you.
Just except small things, I'd like to, because I think deadline for AACR abstract is coming, so I mainly work on producing some results from cancer incidence meta-analysis. But can I like share my data set? So could you just give me a feedback on my plan? Like, I actually hasn't produced any solid result as of now. I'm still like planning to how to do it, because this data, because it's the first time to do this kind of research, there are not many studies I can refer to. So yeah, but I'm worried, what if the resolution would be bad again? So but it's not that
If you
Uh,
share to
Scroll,
me,
I
I
think
can.
it's fine. If you don't scroll,
if
then
I don't scroll, it would be fine.
yeah, just share the screen.
Okay, let me just try, but I might have to scroll. Can you see the screen?
Yeah.
So here, author and year of study. For meta regression, I try to collect some basic information on each study, like study type, cohort, whether it's cohort or case control, and gender of population. This is the time interval. So if you look at here, so many studies use different time intervals. So here, Sirhan, they classified their data into 5 years, 5 to 14, 15 to 29, over 30 years. So I try to group all the time interval into two, like less than 5 years and more than 5 years. So I'm coding that as 1 and 2 to classify them when I do subgroup analysis. And then for cancer type, as you recommended, me and Suwon try to come up with 10 most prevalent cancer types. So here, but then there are some minor subgroups, like some studies went too deep, like they classified non-Hodgkin lymphoma all and only included nodes. And some did colorectal, but some did colon and rectum separately. And so many articles didn't actually give me report actual, like how many people develop cancer among transfusion group or non-transfusion group. This study luckily report that kind of specific detail number, but so many other paper didn't have that number, but they do have estimate of risk. It could be risk, relative risk, or standardized incidence rate, or I'm sorry, I'm scrolling down, but or odds ratio, or
Oh, is that,
all these. Yeah. Sorry, did you say?
no, you were frozen for a second.
Oh, okay, got it. So my plan as of now is, because I read somewhere that if that's the only case, and if that's reasonable, it's still possible to sum up different types of estimates of risk. So I'm trying to pull up these risk estimate of risk, not the actual number, because so just only one paper report that actual number. So I would rather using this kind of relative risk or any other estimates of risk into the meta. And do you agree with that? Or, I mean, it would be possible, but it would take long for me to reach out to each author to give me the actual number of data but
Hmm.
and it because they didn't give me extra number, it's hard for me to sum up data based on different time points. So let's say if I want to look at transfusion risk, cancer incident, risk of cancer developing after 5 years, I would, for one paper, I would have to include 3 different time points in just one meta, because these 3 time points all indicates the risk after 5 years of transfusion. So
Yeah, I understand that. So you're trying to pull incidents?
pull this risk, estimates of risk, incidence, and this, I think, risk of cancer incidence, these values are representative of risk of incidence.
And you're trying to do meta-analysis, pulling the risk or the incidence rate.
Yes,
Some people,
so
I mean, every paper will report that, but
right.
among transfused group, and so for Dr. Cha, this project is looking at, is there a difference in cancer incidence after blood transfusion? And that's been kind of our lab's, focus of interest in the more like the cancer epidemiology. We've published few papers on this already. But this data comes from, I think this is the most comprehensive one, right?
Right.
Because Hyuk-Ju Cho was, this is, I
What
guess,
Seok-Ju Cho, what Seok-Ju Cho did, he only compared whether there's increase of cancer incidence among lung cancer patients.
right, just the lung, and then that was positive, but this is all cancer, and this is all
So
primary
we
incidence.
all,
Yeah.
we collected in total 5 studies,
Mhm.
which reported cancer incidence of multiple cancer types. And
Mm.
then we try to sort them out again, try to pull up each category again.
Yeah.
Yeah.
So you have 5 studies, and you're grouping the cancer type and then pulling the incidence. Now your question,
Yes.
some papers don't report the raw number of like the case and control, right?
Right.
But if we
yeah.
have a total number and the instant, then can't you calculate it?
But like the only for many of these studies, they only reported relative risk of incidence, so it's kind of hard to produce that actual 2 group.
So what do you want to do? I don't have a clear answer for you.
As I looked up some other types of meta, there are some reference that I can refer to. So if there are, I think some articles say that it's feasible to sum up even like different types of estimates of risk. So based on that suggestion, I was planning to sum up the columns here, utilizing these columns to produce pulling up results. Yeah.
I mean, Sirhan, you can do anything, but for other papers, you said you don't.
Yes. And even for Hargream,
Uh-huh.
they don't have, actually they compare the way they calculate this standardized incidence risk is that they only follow up the transfusion group, people who got transfusion, and then they come to produce this kind of a relative risk. They refer to the generalized cancer, risk of general population. So they don't even have a solid compared group.
That's problematic. They don't disclose what the standard at that, yeah, they need to disclose what number they used to get standard.
They just described how they how they come up with this number, and they just said that they compare the risk to the risk of general population at that, at the same year or at the
Mm.
same time period. But they did report risk estimates and with like confidence intervals. So I thought maybe I could use this.
All papers used SIR, then I think you can still pull SIR because you know the total number of patients.
So, but yeah, even though I know the total number of most papers report the total number of patient who or population who receive transfusion, but I think Riddle even didn't report this, but they did report odds ratio. So yeah, data wide, it's not that easy situation to pull up every data, but like I think as of now, the only available way to pull this up is using different estimates of risk.
Well, you can't, odds ratio, hazard ratio, SIR cannot be pulled. They're different metrics.
Yeah,
So-
but then there are some references that support this kind of method.
Oh, really?
Yes, I did found that.
Maybe there's some conversion.
No, we don't need oh, I should bring that paper. Actually, yeah, there was a paper which did this kind of thing.
Why? Yeah, because I don't know the field well. Why don't you study it and do what the paper says it can do, and then present? And
Yeah, okay, I will.
yeah. So yeah, you have my permission to try that and pulling and show us the rationale and show us what what you find if you do that, and we'll go from there.
Okay.
Yeah. But
I thought it, yeah.
But not just the method paper, find the actual paper that used the methods, that is published in decent journal, and show us like an example of how they did it.
Of course, yeah, I could do that.
Yeah.
Okay, so
So that's your cancer instant meta-analysis. Don't you have cancer progression paper?
cancer survivor,
Yeah.
yeah, just a review paper. Yeah,
That one you didn't do meta-analysis, right?
yes. Just
Where-
just narrative and just make table summarizing all the results.
Where is that paper?
That it has been delayed as well. So I would reach out to Dr. Jian if she already gave us comments, but I'm dealing with that comments still. So
I mean, if you're ready, you don't have to present now, but
right.
I just want you to work on
Yeah,
it.
I will hurry up.
Because this has been sitting too long.
That's true. But okay.
So Dr. Seok-Ju Cho is still the first author.
I think so. We were kind of, yeah,
Yeah.
here I think he put these things.
So he's still, whether it's equal, he has to contribute still when revising and submitting. So make sure he does his part.
Okay.
don't think it's you're not the only one to that has to work on this.
Mm,
So
Okay, then I'll, yeah, I
and
think he,
firm deadline and present. This is
Mm.
time for you to invest and finish.
right,
Okay,
right.
make
Yeah.
this a priority in your life. Okay. There
I
are
know.
other competing things. I know you have to make money, you have to meet family, you have to do other things, but
Mhm.
this cannot be delayed. I don't want you to go to residency and say this project has been delayed. Okay,
That's,
you have to be motivated enough. And you've put, your time and effort into this. So
right, right.
close to the finish line, work on it. If you need help, reach out to the other authors, talk to me. Because this has to come out. This is kind of the basis of the whole story.
Okay, okay, I'll, Yeah.
Miss Lee, I don't know what she's doing. She's not doing residency right now. So reach out to people who are co-authors and make them work. You are the project lead right now. And if you think you've worked more than Seokju Cho, your name has to go in front. So the order of authorship is all decided based on what is your investment and who is the final person who submits the paper. If you cannot be the first author, if you're like, well, I'm done with it, revision and submission, other people can do it. Well, you can't be the first author. First
Mhm.
author is who starts and takes it all the way. Okay, I think
Right.
that should be clear.
Yeah.
And Min Jung Kim has written our lab policy for authorship. And
Yeah,
share that with other members.
okay.
Okay, anything else?
I will work on MRD as well through this week. I definitely need to see the epic a lot. So I'm trying to make time with Dr. Jisang or Jian if during this.
So,
Yeah, other than
so
the
where is MRD paper right now? What are we waiting?
MRD paper, we are working with Natera folks, and Natera reached out to us to help us to make a really nice figure, like swimmer plot. They're still drawing swimmer plot for our paper, and then they asked me to give some more further detailed information about patients, and I had to figure that out. I will just summarize all the questions that I received and then give it to Dr. Jisang or Jian.
So the MRD paper did Natera. So Dr. Cha, this is our own Northwestern lung cancer patients. We with Natera, and we're kind of doing like more like a, like a patient data analysis. Yeah. So did Natera add more patients, or what is there anything new since the last abstract we presented?
Yes. They did find some more patients on their side, and then they asked us to give them like clinical data.
Mhm.
So in total, there will be 38 patients, which is way larger than the previous population. So
whatever the table or the graph they make with the additional data,
yes,
you should submit to AACR.
AACL as well. Okay, so they will.
So was it submitted to CISI?
It's been submitted to CC, but that's not the final data set.
So
They
they
added-
added more data, right?
Yeah.
And they made more tables.
Not exactly table, but the swimmer plot itself.
Streamer plot. So show me what they are when they get back to you. Tell them with additional data, tell them we have deadline for AACR. And I know
Okay.
it's not working on it, correct?
I didn't hear you.
Me is not working on this anymore.
I think she's been so busy, like, to get back to me.
Okay, so Seungmi cannot make the abstract, correct?
Mhm.
So Eugene, you should be in charge of making the abstract for the AACR,
For MRD,
for MRD.
that shouldn't be a problem because what I have to do is just modify the 60 version based on the changes
Yes,
because yeah.
but you will be the first author because Seungmi is not working on this abstract.
Okay, so got it.
Okay.
And
So show me so push Natera because tell them we have a deadline. We want to write this paper up ASAP where we want to present at the AACR.
okay,
Okay, and,
Yeah.
and who's writing this manuscript? Who's involved?
I wrote until the results part, introduction, methods, and results. And discussion is done by Seungmi partly, but it's not complete yet. So
Is Seungmi gonna complete it or are you going to complete it? Who's the
I'm trying to complete it.
you
So
said
the discussion.
you are going to complete it?
Mhm.
Okay. If you need help, talk to Arnie and Jian or Liam.
Okay.
They all know the
Where-
problem very well. So you can
Okay.
divide and conquer. We have
Mhm.
other undergrads. If you need help with table referencing, I want you to, because Seungmi is supposed to be the project lead. She's not. So you are the project lead. You have to assign. I think Jui's been doing very good job assigning and pushing. So you should do what Jui's been doing. Assign, push, push. Don't make it all on your plate. It will be all delayed. Okay,
Right.
okay. So I mean, I think it's just been. I thought when Seungmi was leaving our lab, we have the final product, but we didn't. So it's the same thing with other projects. I think we have to push to finish out what we already have and not start something new. Okay. Anything else?
That's all as of now.
Okay. Very good. Very good. Thank you. We'll move on to Dr. Jian.
Yeah. Let me see my screen. Can you see my presentation?
Yeah.
Okay. Well, let me briefly review about the tumor infiltrating lymphocytes in breast cancer. I still studying, but I want to share the knowledge with you. So, well, as you know, the TIL is a very important factor in cancer, and recent study shows that the TIL is also important in the breast cancer, which represents a favorable prognostic factor. And the TIL's effect is similar in the neoadjuvant setting and adjuvant setting as well. After neoadjuvant chemotherapy, if the TIL is remained in the residual disease, that it shows the favorable outcome in breast cancer. And there was a big study, even if it was retrospective, but they included more than 300 triple negative breast cancer, and the patients were receiving neoadjuvant chemotherapy. And when you look at this, the patient group was divided in the high TIL group and low TIL group. And definitely high TIL group showed better survivor outcomes with a statistical significance in metastatic free survivor and overall survivor as well. And I don't know why the PCR was so low rate, but anyway, there was more than 293 patients had residual disease, even in triple negative breast cancer. But when this patient had a YP stage 1A, they did not show the statistical significance between low TIL group and high TIL group. But there was a higher tumor burden even after neoadjuvant chemotherapy. Then the statistical significance came out. So I think the TIL is important in the advanced stage of breast cancer right now. And there was another study. It was pooled analysis study.
What was the regimen, chemo regimen on that group? This group?
Oh, sorry, I can't remember, but because these patients were gathered from 3 different hospitals, I think the regimen was so various.
But this was before
Mhm.
Pembro, right?
Yeah. Before, yes, it was 2014.
Hmm.
Mhm.
And the way they measured TIL is just
It was just by pathologists, not AI powered.
and it's just yes or no, like what how did they classify?
Oh, just yes or no. Yeah,
Okay.
very simple. Yeah. And there was one other study. This is a pooled analysis study. And when you see this article, I could learn about the TIL distribution. When you see figure B, it is less than 5% of TIL. And figure C shows 50% of TIL. And figure D is 75%. And figure E and F shows heterogeneous slice, but it is over 35% of TIL. And E shows only 10% in the stroma TIL, S TIL. And F shows 40% of the stroma TIL. And when you look at this graph, mostly, the gene signature and the breast cancer subtype is not 100% match, actually. So when you look at the triple negative breast cancer, HER2 positive breast cancer, when they show the gene signature as a basal type, ERPR negative or HER2 enriched triple negative breast cancer, the TIL is way more higher than the other types. And when you look at this, definitely high TIL shows better survivor outcome. And interestingly, in luminal type, there are not statistical difference between 2 groups. So mostly the study was focused on the HER2 positive and triple negative breast cancer. And when you look at the figure D, nodal status is positive, which means higher stage, it shows a stronger correlation with the TILs and the survivor outcome. And this was very interesting. You can see 4 graphs, but definitely high TILs with the trastuzumab shows a better survivor outcome, but low TIL with the trastuzumab and high TIL without trastuzumab, these 2 graphs shows a similar. So, well, the TILs is really important for the survivor outcomes, but we don't know this really act like promote the therapeutic effect, whether or not, but it is still important, even the patients who did not receive the trastuzumab.
Well, I'll go back. The HER2 enriched and luminal HER2, is that luminal like HER2?
Yes,
Triple-
luminal HER2 means a triple positive.
Yeah, and in their high-low TIL pertuzumab is different than so if you have luminal HER2,
Mhm.
even if you get trastuzumab, high TIL, low TIL is different here.
Yeah, yes.
Yeah. So hard to, and this is all is this neoadjuvant and adjuvant? Like this group was treated,
It was
all,
uh
okay,
yeah,
not
yeah.
neoadjuvant, like all adjuvant, okay.
yeah, or adjuvant.
Interesting.
Well, the triple positive has a ERPR positive, but they shows a similar graph. So I think HER2 is stronger factor than the ERPR negative.
Go back. The ERPR negative was, if you go back,
What?
I want to see,
Okay.
yeah, luminal.
Uh-huh.
So this is TIL high, low. I mean, it impacts all groups, right?
Mhm.
But more so in node positive and more so, I guess, I guess HER2 positive is less, but some group got trastuzumab, some group didn't. So
Mhm.
that's the old data. Okay.
Yeah, it's the old. Yeah.
Okay, you can
Sure.
move.
Yes. I just studied it to understand the LUNET abstracts, two of the abstracts. So let's look at the abstract one. It was presented in ASCO. And the study aim was to assess the clinical significance of AI-powered partial TIL analysis in the prediction of a PCR in
Mhm.
triple-negative breast cancer. And they included stage 1 to 3 of triple-negative breast cancer. And mostly the immune status could be classified as a 3: immune inflamed, immune excluded, immune desert. So for my understanding, I got one captured webtoon. It came from Roche website. And in yes, in the desert means that there is no T-cell army to fight. And immune excluded means there is T-cell, but they can't enable to scale the walls or cross them at the castle to fight. And immune inflamed means they're fighting and they can win to the cancer enemy. So it was briefly understanding. So came back to the abstract and they measured all of the TILs by AI powered, and well, this is important result. Oh, well, there was so many errors in posture because it should be ITIL, not STIL right here. But
Oh,
yeah, yeah. Anyway, I couldn't understand with the description. So when you look at this ITIL and STIL, mean ITIL score was significantly higher in patients in PCR. So when the patient has TIL, a higher TIL, then the PCR rate is higher with a statistical significance.
Mhm.
And this study was about the triple-negative breast cancer.
Mhm.
So the conclusion was AI-powered partial T-cell could predict the PCR rate well. So, and the second one was presented in ESMO.
Back.
Go back over.
So I don't know why they didn't, because they usually, if you look at our American Journal of Pathology paper, it's 3 groups. I was hoping the inflamed excluded desert
Mhm.
and excluded has more, I guess, TIL in the stroma. But I guess even excluded group, the stromal TIL, having that versus desert did make a difference. I guess that's why the STIL and ITIL were both predictive of better
Mhm.
outcome. So I think that's, inflamed versus
Mhm.
excluded, and excluded versus desert. I think that's why both ITIL and STIL was still a predictive factor.
Mhm.
And just the fact that these three still like separates nicely in triple negative, I think it's just I feel like it's real. This is not something like randomly found.
Mhm.
And this is the RFS, and then PCR rate. There seems to be like a dose response, 14, 20, 17's kind, but still, yeah, I think this is real phenomenon. And in triple negative, we kind of knew TIL was important, but to quantify it this well and make it into these three top categories is the first, they've done. So
Yes, this two study was performed with the Samsung and Asan data with the patients before 2015. So they couldn't get a recent current treatment like a pembrolizumab or pertuzumab. So that could be our new perspective in our study.
oh,
Which
even for
means that
do
yeah. I will show you. So this is abstract 2,
okay.
and the patients it was included in 2009 to 2017. So probably the patient cannot receive a pertuzumab at that moment for immunogenicity therapy. Anyway, the population was high, for more than 1,000. So it was presented in ESMO. It was so similar as before one. And when you look at the HER2 categorization, and the AI-powered HER2 was very well matched with the research by the pathologist manually. And so this is a strongest, I mean, they want to show this one from the company, I think. But this is our interesting, not before one. So when you look at this, when the higher stromal TIL, higher intratumoral TIL, then the PCR is way more in the tumor. So the conclusion was so similar as before one. AI-powered automated HER2 scoring is very accurate, and this can predict the PCR. But these are all retrospective, and as I mentioned, the patient couldn't get the current treatment or new drugs. So there were several other papers with the deep learning TIL to see in the various cancer, but these were very similar as the LUNI did. And actually in breast cancer, TIL area is lower than the other cancers. You can see the stomach cancer and lung cancer and thymoma, very higher TIL area they show. That in breast cancer, still the TIL area is lower. But even it is lower, but HER2 in triple negative breast cancer TIL is important, I think. So I just want to move on the our study. So this could be our study. We can just combine with the ctDNA result, or we can make another abstract because the patient population is so they receive the current treatment with the pertuzumab. So this could be different thing with the previous LUNIS one. So that's my opinion. So we'll think about it.
Any comments from Dr. Cha?
Actually, I'm not much knowledgeable about the breast cancer and also tear. Yeah.
So Dr. Jian has her own neoadjuvant breast samples,
Yeah.
tissue and blood before, during, and after neoadjuvant therapy. So
Yeah.
then with the tissue, we will analyze the TIL,
Mm.
and then from blood we have ttDNA dynamics,
Yeah.
PCR rate. So I think we're gonna look at it for the first time. It's good to know no one has looked at it yet.
Well, yeah, I
So
want to.
where are we? Are we like with the, I mean, the logistics? Are we are we gonna ship it to Lunit first, or are waiting for IRB first? I guess is that.
Oh, no, I asked to pathologists to get the digital images
Yeah.
directly, so I'm waiting that.
Oh, okay.
And I think for the predecessor study, the sample is ready, but I need to discuss with you. So can you see my Word file right
Yeah.
now?
Mm-hmm.
Okay, so this is my update.
Mm-hmm. So
So
yeah. Yeah, go ahead.
First project is double lung transplantation for bilateral lung cancer. So we got the data from UNOS, and I got the reply last night from the international registry, finally. So I'm so excited to make a table. I think we can make a table 1 with the case reviews, and the table 2 with the UNOS data, and table 3 with the international registry.
Yes.
So, well, Dr. J did really great work with the UNOS data, so I could analyze it.
Mm.
So this is a graph that includes only cancer, but it is only 15 cases. It was so simple and looks be so poor. So I included the other non-cancer registry. It is more than 30,000. But I could get the graph from them. Definitely there is a statistical significance. Yeah. How do you think about this graph? It is a UNOS data.
So cancer versus
Yeah, I know. It's
yeah, I mean the UNOS, like all lung cancer, other lung cancer transplant indication, right?
only lung cancer. And these were all BAC.
All BIC, like in
Yeah.
15.
Yeah.
All BIC. And what,
Yeah.
all double transplant?
Yeah, all double. We are focused on only double.
Yeah. So all, wow, 30,000, that's a lot of non-cancer. So I don't think any data like this has been presented. Were there?
No.
I think it's still worth presenting.
Oh my God.
I want to ask you about the population included in the study. The 15 patient, what is the inclusion criteria for the 15 patient?
15 patients received the double lung transplantation who had diagnosed as lung cancer. Mostly for the organ transplantation, most of the candidates shouldn't have cancer before transplantation, generally.
Right.
But for these lung cancer patients, they got double lung transplantation, and we could analyze the survivor. And I found the after 2000 to currently, most of the lung transplantation was performed to the non-cancer patient. That's why we couldn't find it more than 15. There
Mm,
were all the patients within that period, but they were not cancer.
mhm. Well, I have a question. So were they transplanted to treat lung cancer? Or is anybody had incidental lung cancer?
Not incidental lung cancer.
They got the transplant because
Yeah,
of,
yes, yes,
Okay,
yes, yes.
that has to be
This
made,
is a past lung disease,
yeah,
was lung cancer.
because
It-
they had the lung compromise because of lung cancer.
Yes, because of lung
So I think we should make that clear that
cancer. Mhm, mhm.
paid for transplant was
Mhm.
because of lung cancer bilateral.
Exactly.
So all these patients have residual lung cancer at the time of lung transplantation, and
It-
without evidence of other cancers or other.
Actually, we can't get the information. This is a registry from the
yeah,
US
yeah.
government.
Yeah.
They
And-
gave us
yeah.
just the survivor
Yeah.
or a very brief information,
Yeah,
but we couldn't get
Yeah.
the
And-
very
okay.
detailed.
Yeah, but I think we can assume that there is no metastatic.
Mhm,
Otherwise, they
mhm.
don't proceed with.
Yeah.
you know,
Yeah, and can I see the median survival time?
Median
What is the
survival.
median survival time, and is there any long-term survival? There is more than
No. Well,
median-
there is a survivor.
So this is quite interesting. Actually,
Yeah,
we also have an experience of liver transplantation for
Mhm.
liver limited metastatic cancer. Yeah,
Yeah.
but
Can you see my axon?
yeah,
Can you see my axon file?
yeah.
So these are all we have, all blank, all blank.
Yeah.
Yeah. But
But
fortunately, we could get these survivors. So mean is 27 months.
Yeah, 27 months,
Yeah,
66.
that's the mean, not median. Okay. Okay. Yeah, I think we need to calculate.
But in most patients, the cause of death is not related to lung cancer.
Yeah.
No, interesting. Yeah.
Yeah.
I want to make it in the table.
Yeah.
So I'm working on it. Oh, not in here. Okay. I try to make one table with the UNOS data because it is only 15. So I want to present all case numbers and the cause of death and survivor. It could be interesting.
And I was surprised that 30,000 patients have underwent bilateral lung transplantation.
Yeah, interesting.
Oh,
Yeah. It's the
wow.
US. Welcome to
Oh.
US.
But it is very surprising that the survival between the groups is so different when the most probable cause for death in lung cancer group is not related to cancer.
Mhm.
So
Yeah, I think small sample size.
yeah.
Yes,
But
it is
yeah,
not reliable.
yeah.
So for non-cancer group,
Uh-huh.
all have cause of death column, right? So
Yes.
because people talk about immune suppression and then you can get malignancy because of immune suppression. Of course, you can get infection
Mhm.
rejection,
Mhm.
but infection rejection is common cause of death.
Okay.
But it would be good if you can draw
Oh,
cancer specific survival. between the
Uh-huh.
groups. It might be
Okay.
Yeah,
Dr.
cancer
J,
specific survival. Yeah,
Dr. J, I need, I need you.
yeah.
Because he can sort it.
so Dr.
It was
Yu,
all coded. So
yeah.
we have to solve the puzzle.
So I have one question. So the previous Dr. Charles said that if there is patient with the previously diagnosed cancer,
Mm.
right? So you think the previous diagnosed cancer might affect survivor, right?
야, 잠깐. 여기서 한국말로 잠깐 진행합시다.
어,
영어 하시는 선생님들이 지금 없어서 괜찮습니다.
네.
오케이. 네, 알겠습니다.
네.
네.
편해지네요.
아니,
네.
선생님 나가셨군요.
네.
네, 일단 넌 캔서 그룹에서 그러니까 그 이전에 이제 진단받았던 캔서를 그러니까 예전에 이 리시피언트가 예전에 진단받은 캔서가 있는지 없는지 그런 거는 다 확인을 할 수가 있거든요. 혹시 그것도 하나 만들면 솔팅을 하면 좋을
있었는지 없었는지.
네.
네, 근데
런 캔서가 아니면 상관이 없을 것 같은데요. 여기서는 오히려 혼란만 일으키고, 왜냐면 런 트랜스플란테이션을 함에 있어서 다른 캔서가 오펙트를 할 거는 없을 것 같거든요. 근데 그렇게 되면 우리가 포커스 하려고 하는 데서 벗어나서 너무 방대한, 정말 유노스 데이터를 애널라이시스 하는 그런 페이퍼가 나와야 될 것 같아요.
네, 그냥 cause of death가 캔서 때문인지 아닌지만 보는 걸로 해주세요. 네.
네, 이게 지난번에 유노스 데이터에서 봤던 건데요. 근데 사실은 이 사람들은 싱글이랑 더블렁을 같이 봤거든요. 그래서 좀 비슷하긴 한데 저희는 더블렁만 지금 뺐고 이게 2012년이니까 10년간의 데이터가 더 축적이 된 거예요. 저희는.
아,
좀 그거에 차별성이 있을 것 같습니다.
그러네.
그리고 이 페이퍼는 유노스 하나만 가지고 리뷰를 했고요.
네.
저희는 지금 이 리뷰 페이퍼 안에서 진짜 테이블 하나, 피겨 하나 딱 여기만 유노스를 쉐어를 할 생각이어서 훨씬 더 큰 리뷰 페이퍼가 나오지 않을까 생각합니다.
네, 좋은 것 같아요. 근데
그리고
이거 레슨은 BAL 브론키오 알베올라 패턴이든 아니든 지금 큰 차이가 없다라는 건가요? 이 그래프는?
네, 캔서 페이션트든 아니든 이 서바이벌 아웃컴이 비슷하다고 이게 제너럴이 이게 여기도 2만 명이 넘잖아요.
네.
넌 캔서로 렁 트랜스플란테이션을 받았던 그룹이거든요.
근데 왜 우리 데이터는 차이가 많이 나죠? 싱글이
이게
빠져.
지금 네네 10년까지 왔거든요. 여기는.
네.
그래서 10년까지 오면 여기쯤에서 사실은 저희가 끝이 나요.
네.
그런데 저희는 사실 더블만 보기도 했고 그리고 훨씬 길어지면서 캔서는 이미 환자들이 다 익스파이 해버렸고요. 근데 앞에 레퍼런스랑 크게 비교할 필요는 없을 것 같아요. 왜냐면 10년의 데이터가 더 축적이 됐고
네.
더블렁을 받은 사람은 바이래트럴 렁 캔서를 가졌기 때문에 한 거기 때문에 훨씬 더 서바이벌 아웃컴이 안 좋을 수 있다고 저는 생각을 합니다.
네.
근데 제가 잘 몰라서 그런데 이게 바이라테럴 렁 캔서가
네.
그냥 이게 그러니까 선생님 예전에 그 수술하셨던 그 환자분은 이제 메타스타틱인가요? 아더 사이드가.
그러니까 그 브론크에서 림프노드를 다 해봐도 하일러 림프노드 다 해봐도 없어요. 그냥 오로지 에어웨이 통해서만
네.
그런 환자들만 저희가 수술했어요.
그러니까 세임 히스톨로지인 거.
히스톨로
그 차
네.
선생님 말씀하신 게 뭔지 알 것 같아요.
네,
제가
네.
처음에 궁금했던 게 스테이지
네.
4인데 왜 바이래터럴이냐고 생각을 했는데 바이래터럴이면 스테이지 4더라고요.
네, 네, 네.
다른데
네,
메타가
네.
없어도.
예, 근데 이제 사실 그러니까 제가 좀 궁금한 거는 그러니까 바이라테랄이 이제 사실 메타스타틱이잖아요. 프라이머리 튜머 이쪽에서 다른 쪽으로 간 건데 이제 그런 경우 말고 그냥 정말 인시던털하게 싱크로너스인 경우도 좀 있나요? 사실 제가 잘 몰라서 렁 캔서는요. 그런 경우를 생각할 수 있는 여기 이제 그런 환자들이 얼마나 포함됐을 수도 있을지 이제 그런 걸 생각해 봐야 될 것 같긴 한데 뭐 사실 콜론은 그런 게 워낙 흔하니까요. 싱크로너스 캔서가. 근데 이제 렁은 제가 잘은 몰라서 그래서 뭐 예, 그런 걸 좀
양쪽 폐에 싱크로너스하게 더블
그렇죠.
캔서가
그렇죠.
생기는
예를
걸
들면 히스톨로
말씀하시는
그렇죠.
거죠.
히스톨로지가 좀 다를 수도
네,
있고
네,
아니면 뭐 스테이지 1, 스테이지 1 이렇게 뭐 정말 얼리 캔서 두 개가
네.
이렇게 있을 수도 있고 그래서 그런 부분이 네,
있을
좀
여기서는 아마 없기는 어려울 것 같아요.
그렇죠. 그런
이게
거 없을 수는 없을
네네.
수
왜냐면
없겠죠. 네, 네,
이식
네,
대기를 하기 위해서
네,
레지스트리에 먼저 등록을 하고
네,
그리고 등록을 한 만큼
네,
나중에 아웃컴을 추가로 등록해라 이렇게
네,
레코멘드를
네,
하는데
네,
사실 아시잖아요. 수술
네,
끝나고
네, 네,
나면 레지스트리 등록
그렇죠.
잘들
네,
안
네, 네,
하시는
네,
거.
네,
그래서 아마 뒤쪽에
네,
데이터들이 조금 빈약할 것
네,
같고요. 거기까지는
네,
저희가 오기가 어려울 것
네,
같습니다.
그리고 몰라요. 두 개의 프라이머리였는지 이게 메타가 돼서 만대 쪽으로 갔는지 뭐
그렇죠. 예,
어디가 어려워.
하기 쉽지가 않아요.
네,
네.
근데 대부분의 BALC는 그 스타스라고 spread through airway로
네,
되는 경우가 대부분인 것 같아요. 그리고
네,
되게 어그레시브해요.
네,
그래서 히모 이뮤노 타겟 테라피
네,
저도 다 안 듣고요.
네,
그래서 같이 그냥 같은 패턴으로 그냥 림펜제릭 스프레드 같이 그냥 쭉
네,
퍼지면서 그냥 돌아가시기 때문에
네, 네, 네,
퍼지는 것 같아요.
네, 네,
보통 싱크로너스는 이제 하나는 좀 인돌런트하고 뭐 하나 뭐
네,
패턴이 다른데 보통 말리그넌트하게 이게 퍼지는 패턴이 되게 비슷해서 저는 그냥 대부분 비슷한 것 같아요. 하나로. 네.
네. 이 15명은 다 BAC인
네, 네,
거죠? 네네.
그렇더라고요.
네네.
네. 그러니까
네.
다른
그렇죠. 네.
히스톨로지에서는
네.
아예 시도를 안 하는 거.
그렇죠. 그럴 것
네,
같아요.
네,
그 어떻게 보면은 저희 케이스 리포트가 저희 병원께 BAC 쓰고 BAC가 아니었잖아요.
네,
그런
무시너스 아데노크라신.
게 잘하고 있어서
네, 네,
저희가 이제 프로포즈 하는 거는 다른 타입에서도 그 림파틱 블러드스트림의 에비던스가 없으면 해도 되겠다라는 거죠. 저희가 주장.
네.
그래서 이제
나아가서
인터내셔널
네.
우리 차용준 선생님이 하셨던 것처럼
네.
DNA, RNA
네.
봐서
네.
what
네,
feature
네.
또
네.
네,
네. 네.
로컬리만 키워
그렇죠. 네.
가능한 피처가
네.
뭐냐
네.
그거를 저희는 이제 템퍼스라는 회사랑 같이
네.
Hey Liam, where are we with that Tempest proposal?
They haven't gotten back to us yet. We've sent them follow-up emails, still no reply.
한 번 더 팔로우 해주세요. 저 CC에서.
네.
너무 좋은 것 같아요. 프로포즈한 게 작년에 AAC 할 때 프로포즈 했는데, 그러니까 올해 봄에 AAC 할 때 프로포즈 했는데 너무 오래 걸리는. 예. 오케이.
네.
예.
그래서 차영준 선생님께 하나 더 말씀드리면 리커런트 패턴을 본 스터디가 하나 있었는데요. 그거는 다른 사람의 폐를 이식을 받았음에도 불구하고 리커를 하면 프라이머리 캔서랑 그 진 시그니처가 동일하게 그러니까 결국은 캔서가 레시피언트의 렁에서 그 원래 갖고 있었던 캔서랑 동일한 캔서가 생기는 걸
네.
봤던 스터디는 있었다. 네. 그래서 테이블 3는 지금 인터내셔널 레지스트리로 지금 구성을 할 생각이고요. 여기서 어젯밤에 연락이 왔는데 이분은 퇴근하고 항상 이메일을 주시는데 2000년에서 2018년까지만 데이터가 자기들이 있는데 괜찮냐 그래서 그걸 달라고 했고요. 그리고 멤버는 없어도 된다는 답변을 받았습니다. 그래서 일주일 정도 걸리니까 좀 기다려 달라고. 네. 그래서 이건 그렇게 구성하겠습니다. 테이블 1은 그래서 푸드 애널라이시스라고 했는데 사실은 각각의 기관의 선생님들은 그 뒤로는 답을 안 주셨어요. 그래서 그 사람들이 거의 대부분 사실은 페이퍼 안에서 데스까지도 다 디스크립션이 된 페이퍼들이 있기 때문에 그 사람들의 롱텀 아웃컴은 크게 의미는 없겠다 생각이 들어서 저는 그냥 리뷰 아티클들을 다 모아서 새로운 테이블을 만들려고 생각 중이고요.
네.
최 교수님께서 처음에 보여주셨던 그랜빌 그 메인 아티클 우리가 레퍼런스로 쓰려고 했던 그 아티클에는 5개의 그 리뷰 아티클들이 메인 프로젝트다 이렇게 되어 있는데 실제로 수는 적지만 리뷰 아티클들은 그 뒤에도 계속 조금씩 나왔거든요. 그래서 저는 그거를 다 푸드업 해가지고 말 그대로 저희가 타이틀이 푸드 애널라이시스니까 그거를 다 리뷰를 해서 이제 테이블 1을 구성을 하려고 생각 중입니다. 그리고 그 그 리뷰 아티클들 중에서 지금 현재는 더블 렁 트랜스플란테이션을 받은 환자군만 제가 이렇게 계산해서 뽑아내고 있는 상태거든요. 거기는 싱글, 더블 다 이렇게 믹스되어 있어서 우리가 필요한 건 더블 렁이니까 앞에 또 레퍼런스랑 또 차별화가 되어야 하니까요.
맞아요.
손절하면 안 되니까. 그래서 더블만 가지고 지금 제가 서바이벌 레이트를 지금 계산을 해보고 있습니다. 그래서 조금 더 되게 많아질 것 같아요.
근데 이거 중간에 레이오마요sar코마 있는 건 어떻게?
안 그래도 이거 교수님께 여쭤보려고 그랬어요. 이 사람은 메타스타틱, 나머지는 프라이머리인데 이 케이스만 메타스타틱이거든요. 그런데 넣을지.
했다는 게 되게 놀라워요. 사실은 그래서
네.
그 콜론에서 리버 가는 거랑 똑같이 sar코마에서 렁 가는 게 비슷하거든요. 렁에만 주로 가고 렁 외에 딴 데 안 가고 그 다음에 sar코마 프라이머리는 리섹션으로 제거돼서 뭐 그쪽에서 리커하는 건 없고. 네, 그래서 저희가 더블 렁 트랜스플란트에 이런 sar코마를 넣었어요. 그쵸? 리암 선생님 맞습니까?
네, 저희도 그 eligibility 포함되어 있기는 해요.
네네네네. 그러니까 똑같이 프라이머리 렁 캔서도 넣고, 그러니까 저희 지금 얘기하는 거는 프로스펙티브 저희가 병원에서 하는 트라이얼 얘기하는 거예요. 네, 그래서 이런 환자군에서도 이제 아웃컴을 추적하려고 하고 있고요. 하지만 이 리뷰 페이퍼는 좀 다른 것 같아요. 그냥 에비던스 이게 벌써 에비던스가 있는 거잖아요. 메타 해갖고 거기까지 갔다는 거. 네, 그래서 이거를 이 프로젝트를 위해서는 빼는 게 좋을 것 같아요.
알겠습니다.
네. 근데 뭐 아웃컴은 어땠어요? 페이퍼에서? 뭐
그냥 케이스 리포트고요. 한 2년? 2년 정도까지 서바이벌 했다 거기서 끝난 것 같아요.
그래도 2년을 살게 했다면
네, 네, 네,
훌륭하네요. 그 정도면.
네,
네.
네. 나머지는 제가 조금 더 작업을 하겠습니다. 그리고 피겨는 쌤한테 제가 요청을 했고 더블 렁 트랜스플란테이션에 포커스하고
네.
그리고 좀 더 여기 피리어드를 익스텐션 하라고 얘기를 했고요.
네.
다시 2주를 드렸습니다. 그래서 아마 다시 답변을 주실 것 같고요.
네.
라이팅 프로세스는 지금 그래서 자 여기 인시던스까지 아니 저기 그 뭐지 인디케이션까지는 마쳤는데 지금 클리니컬 아웃컴을 작업을 하느라고 나머지는 약간 홀딩이 되어 있고요.
네.
최대한 빨리 작업을 하겠습니다.
네.
그리고 두 번째 프로젝트로 넘어가면
네.
이제 ctDNA in HER2 breast cancer인데 아까 최은강 교수님 말씀 주신 대로 제가 가지고 있는 시리얼 샘플 34명의 환자군에서 128개의 샘플을 가지고 지금 ctDNA를 하려고 하고요.
네.
교수님 다 이거 녹였거든요.
네.
그래가지고 네. 이게 줌이 달라져서 이제 그냥도 화면 공유가 되네요. 그래서 교수님 약간 제가 이 부분에 4cc가 안 되는 것들이 몇 개가 있어서 사실은 네, 토탈 볼륨을 보시면 될 것 같고요. 128개 중에 한 30개 정도가 4cc 가까이는 되는데
네.
4cc가 안 되는 것들이 있어서 얘네들은 어떻게 쉐어를 할지 아니면 이 환자 같으면 이렇게 3개는 시리얼이 되거든요. 그러면 이거는 그냥 제외를 할지.
아, 그거 하나 때문에 그 환자는 또 시리얼에 또 제외가 되겠네요.
네네네네, 여기도 또, 네, 그런 식이 돼서. 근데 이 환자 같으면 지금 다이그노시스가 없는 상태여가지고요. 이게 과연 의미가 있나 하는 생각도 들고.
베이스라인이 제일 중요한데.
네, 이게 베이스라인이 지금 3.5밖에 안 돼서. 그리고 굉장히 안타까운 케이스가 한 2개 있는데요. 1. 얼마밖에 안 되더라고요. 왜 그런지 모르겠는데.
베이스라인이? 아이고.
네, 근데 이 환자는 그냥 뭐 피가 많이 안 나왔는지 뭘 해도 다 적어가지고요. 이 환자는 결과 자체가 잘 안 나올 수도 있겠다는 생각도
그러네요.
들어요. 그래서 일단은 저기
어,
프레디슨용으로 뭐 미니멈 4cc를
다 보내야 될 것 같은데, 그 일은 좀 그렇다. 그, 어,
이 환자는 뺄까요? 이게 다 지금 적어요, 사실은.
네, 다 적긴 하네요. 그러게요.
이것도 다이그노시스가 적어가지고.
네, 보통 이제 10cc 우리가 4를 컷오프로 했는데 아예 안 보내기는 되게 애매하고. 근데 만약에 그러니까 이 암이 정말 쉐딩을 많이 하는 암이어서 그냥 얼마만 좀 있을 수도 있고 그건 우리가 알지를 못하니까요.
네,
제안하는 게 일단 4cc가 안 돼도 보내긴 다 보냅시다.
네,
네,
알겠습니다.
보내긴 다 보내고 그 친구들이 해보고 이건 너무 못하겠다 그러면은
네,
안 하면 되고
그때,
우리가 4cc를 못 넘긴 샘플들 환자분을 다
네,
체크를 해놓고
네,
그다음에 나중에 센시티비티 어널리시스 할 때 4cc를 넘기는 환자들과 함께 또 해보고 못 넘긴 환자들과 빼고도 한번 해보고
예,
네, 그런 식으로 한번 해보고요.
네.
그 프로티오믹스를 할 때 최소한의 양을 각 컴퍼니별로 뭐라고 했나요?
네, 교수님, 그거 제가 뒤에서 다시 말씀드릴게요. 네, 그래서 일단 그러면은 프레디슨 스터디를 먼저 정리를 하면 샘플은 지금 준비가 다 됐고요. 근데 지금 제 마음 같아선 IRB 통과 전에 보내면 어떨까 생각 중인데 이게 궁금한 게 파톨로지스트의 H&E 슬라이드랑 동시에 꼭 가야 되는 건지 아니면은 어차피 우리가 디지털 스캔 이미지를 받을 거니까 샘플만 먼저 프레디슨으로 보내고 디지털 스캔 나중에 받아서 그렇게 해도 될지.
네, 그러니까 제 생각에는 프레디슨에 물어봐서
네,
너네 H&E 슬라이드 보내달라는 게 투머 퓨리티 보려는 거니까 이미지만 보내겠다. 슬라이드가 아니라 그걸 걔네들이 뭐 녹여서 할 거 아니니까.
네, 네,
네, 괜찮냐? 꽤 계산의 해상도의 이미지다. 괜찮다고 할 것 같아요. 그러면 또 그걸 보낼 필요 없을 것 같고 블러드만 보내도 될 것 같아요.
네, 네, 알겠습니다. 제가 여쭤보겠습니다.
네.
그래서
교수님.
네.
죄송한데 교수님 12시에 암전 미팅 있으시거든요.
그렇다. 요거까지만 일단 끝나고 미팅 가겠습니다.
네, 나머지 할까요?
네, 요거 한번 5분 안에 끝낼 것 같죠?
네, 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 맞아요.
끝나고
네, 네,
얘기할게요.
네. 네, 루닛 스터디는 그래서 여기 패널로지스트가 디지털 이미지를 주시면 루닛으로 보내볼 수 있을 것 같고, 앞서 말씀드린 것처럼 펄투스맙을 받았던 환자분이라는 점, 그리고 또는 ctDNA랑 컴바인해서 애널라이시스를 해볼 수 있겠다는 점에서 차별화를 만들 수 있을 것 같고요. 세 개의 컴퍼니는 인터벤은 아직 답변을 못 받았고요. 볼티스는
잠깐만.
네.
인터뷰에 관심 없다고 그랬나요? 리암 선생님? 관심
네,
없다고.
인터벤이 저희 바이오마카 프로젝트는 안 한다고 연락 왔고요.
아, 그럼 괜찮습니다.
네, 그리고 버티스랑 바이오데식스 그리고 델핀은 원래 알고 있었고요. 그래서 연락 받았습니다.
네. 버티스는 150까지도 가능하다. 세이플리 500. 네. 버티스는 또 200, 바이오데식스는 250. 네. 그래도 생각보다 네, 만약에 두 컴퍼니를 250으로 통일하면 0.5가 나가는 거네요. 네, 250으로 통일합시다. 바이오데식 버티스는.
그럼 여기를 먼저 쉐어하고 나머지를 델피를 보내요.
델피는 얼마라고 그랬죠? 아까?
델핀은 800 마이크로리터입니다.
네?
800 마이크로리터.
그게 미니멀이래요?
네, 그때 델피가 얘기했을 때 그렇게 얘기했어요.
네, 다음 분.
꽤 되네요. 그러면 이렇게 합시다. 우리가 4cc를 마지노선으로 했으니까 4cc가 넘어가는 아까 보여준 표 중에서 4cc가 넘어가는 샘플이 있으면 네, 버티스 250 정도를 뺄 수 있으면
네,
빼면 좋겠네요. 만약에 0.5 정도가 여분이 있으면 250씩 두 개를 빼면 되고 그렇게 한번 생각해 보고요. 만약에 그리고 다이내믹스를 보지만 제일 중요한 건 베이스라인이기 때문에 만약에 다른 게 다 없어도 베이스라인만 있으면 베이스라인이 4.5cc가 있다. 그러면 그걸 다 빼서 베이스라인만으로 프리딕션 하는 연구는 할 수 있으니까 그렇게 0.5가 나갔을 때는 두 개 컴퍼니 걸 빼고 그렇게 진행하면 될 것 같아요.
교수님 델피가 프로테오믹스보다는 프라이어티가 지금 현재 있는 거죠. 저희가
네,
나누는 데 있어서. 알겠습니다.
그러니까 0.8이 있으면 또 0.8을
네,
프라이어티는
먼저 빼.
3번으로 할 거예요. 네, 프라이어티는.
알겠습니다.
그래서
그리고
베이스라인으로 충분하면 하고 그 다음에 이제 다이내믹하게 다른 것도 충분히 있으면 뽑고 하지만 4cc보다 더 내려가지는 않게끔
네,
네 그렇게 해서 이 코호트에서는 몇 개 못 뽑더라도 그래도 뽑는 게 나으니까
네. 지금
네,
새로운
네.
코드를 다시 지금 작업하고 있어가지고요.
네.
그건 제가 한번 알아보겠습니다.
나머지 컴퓨터들은 컴퍼니들은 그쪽에서 더 많이 피를 가져가도록
네,
네 그렇게 프라이타이제이션 한번 해봅시다.
이상입니다.
그런 것 같아요. 네. 네, 다른 진아 선생님이랑 리아 선생님은 오후에 프로젝트 저랑 얘기하시고요. 랩 미팅은 일단은 오전 랩 미팅 여기서 끝낼게요. 용준 선생님 오늘 참여해 주셔서 감사하고
네, 너무 많이 배웠습니다.
네,
감사합니다.
우리 하는 프로젝트가
네.
워낙 많아가지고
네.
사실 그것도 있거든요. 그 블러드랑 티슈에서 DNA 헤테로지네이티 분석하고
네.
그다음에 티슈 블러드 TMB 이제 그런 것도
네.
연구하고
네.
그쪽도 많이 하려고 하고 있어요. 그래서
네.
자주 들어오셔서
네.
인풋을
네. 제가 많이 배우는 의미에서 많이 들어오겠습니다. 네.
네, 저희도 또 그쪽에서 하시는 또 랩에서 하시는 연구도 많이 배우고 그러면 좋을 것 같아요. 네, 하여튼 오늘 와주셔서 감사하고 저희 랩에서
감사합니다.
또 여기 계시는 세 분 선생님은 다 여기 지금 저희
네.
랩에 있거든요. 그래서 또
예.
놀러 오셔서 같이 식사도
예. 예, 그럼 좋을 것 같습니다. 네.
이제 진짜 만나서 또 얘기하고
네.
하면 또 좋을 것 같아요.
예,
시카고에.
예,
네. 그래서 또 언제든지 놀러 오세요. 네.
네,
또 다음에 뵙겠습니다.
네네, 감사합니다. 네.